Good morning. Welcome. We will, this morning, if I could have the next slide, please. Do we need to do introductions again? Okay, I'm sorry. So we do need, in addition to, I'll go over the format in a moment, but first we will go around the room and introduce ourselves. I'm Dave Eaton, serve as the chair of this peer review panel, and I'm from the University of Washington.
Chad Wystone, NTP, NIEHS.
Dave Malarkey, NTP.
Michael Wyde, NTP.
Amy Briggs, contractor for NTP, NIEHS.
Keith Shockley, NIEHS.
Christine Witt, NTP, NIEHS.
Wolfgang Kaufmann, retired director of pathology at Merck.
Susan Felter, Procter & Gamble.
Rick Adler, I work for GlaxoSmithKline.
Mark Klein, Wake Forest School of Medicine.
Frank Barnes, University of Colorado.
James Lynn, University of Illinois, Chicago.
Asimina Curti, Ohio State University.
Larry Whiteley, Pfizer.
Matthias Rinke, retired director of pathology from Bayer.
Kamala Pant, BioReliance, Millipore Sigma.
Jack Harkema, Michigan State University.
George Corcoran, Wayne State University.
Lydia Andrews-Jones, Director of Pathology, Allergan.
Tyler Malice, Data Management Services.
Mary Wolf, NIHS NTP and Designated Fellow Official.
John Bucher, NIEHS NTP.
Galen Kempke, NIST.
Matt Ply, FCC. Gonçalo Gamboa, FDA Liaison Officer to the NTP.
Edmond Jerrison, Food and Drug Administration.
Kelly Shipkowski, ICF. Camden Bird, ICF. Jeannie Liu, ICF.
Carl Blackman, Wake Forest Medical Center.
Don Stump, Charles River Laboratories, and representative for the NTP Board of Scientific Counselors.
Gabrielle Wilson, EPL.
Russell Owen, U.S. Environmental Protection Agency.
Ron Melnick, retired NTP, NIEHS.
Shubert Churchill, contractor for NTP.
Peter Little, EPL.
Hiroaki Miyagi, consultant.
Miles Kapsey, ITIS Foundation.
Zurich. Niels Kuster, ETH Zurich and ITIS Foundation.
John Madbury, NIST, RF Fields Group in Boulder, Colorado.
Mark Sesta, NTP, NIEHS.
Katanahali Janardhan, Integrated Laboratory Systems.
Robert Sills, NTP, NIEHS. Maggie Grubel, EPL.
Joe Elder, retired EPA, retired Motorola, and now consultant.
Cynthia Wilson, Integrated Laboratory Systems.
Mario Smith, Social and Scientific Systems.
Grace Kissling, retired from NIEHS NTP.
Alex Merrick, NTP.
Rick Paulus, NTP, NIEHS.
Molly Valland, and NTP, NIEHS.
Nigel Walker, NIHS NTP.
Michelle Huth, NIEHS NTP.
Ruth Lunn, NIEHS NTP.
Georgia Roberts, NIEHS NTP.
June Dunick, NIEHS NTP.
All right, thank you, everyone. Yes, Dr.
I'll read the the conflict of interest statement. Members of the NTP Technical Reports Peer Review Panel serve as independent scientists and not as a representative of any organization. Each member is to exercise judgment as to whether potential conflict of interest might exist for the topic under discussion by the panel due to his or her occupational affiliation, professional activity, or financial interest. This potential for a conflict of interest extends to the member's spouse, minor child, partner, or organization. Panel members are asked to bring to the attention of the federal official any potential conflict, and it will be handled in accordance with departmental policies and regulatory requirements. I'll ask each individual when you're speaking, please use the microphone as the chair iterated, and to introduce yourself. The meeting is being webcast, and it's easier for people watching the webcast to identify who's speaking. If you've not done so, please sign in at the registration desk so we have an accounting of who's in the room. There will be a meeting report prepared after the final day of the meeting, and it will be shared with each of the panelists before it's finalized by the chair to ensure that we captured correctly your comments. The slide presentations from the 3-day meeting will also be posted to the website. They, like the meeting report, will be made accessible for visually and hearing impaired individuals prior to posting. Breaks and lunch are at discretion of the chair. We certainly thank each of you for your service.
Thank you. So this morning we will spend, prior to our break, the morning hearing from NTP staff and consultants about the animal studies. We will then have a break, and after that break we will have the public comments, much in the same format as we did yesterday, in which each public commenter will take the podium, identify himself or herself, and please, for the public comments, limit your comments to 5 minutes so that we have time for discussion and time for everybody to make that time frame. Following that, we'll have a lunch break, and then this afternoon we will hear some additional presentations on the NTP studies in mice. And from that, we'll then follow with a peer reviewer's comments on the study, and then some discussion. So with that, I am happy to turn this over to Dr. Chad Blystone. Chad.
Okay, good morning. I'm going to go over some of the brief elements of the NTP's toxicology and carcinogenicity studies. As I referenced yesterday, as part of NTP's mission, NTP provides data on various agents due to concern of public health effects and for public health protection. Some of these are chronic toxicity and carcinogenicity studies, and these are conducted on agents of concern to identify hazards after long-term exposure. And these studies are described in NTP technical reports, which describe the conclusions as levels of evidence for carcinogenic activity, and the 2 drafts you'll be reviewing today and tomorrow. So I'm going to provide just a brief overview on some of the elements that go into the design of these studies, and also the factors that are used in the analysis and interpretation to help with the discussion today, and then also a little bit on the hazard identification and what we are categorizing as levels of evidence for carcinogenic activity. So the overall objective of these studies is to identify a potential increase in toxicity and or neoplasms from long-term exposure to a test agent. There are several elements that go into the designing of these studies for chronic toxicity and identifying carcinogenic activity. And these include selection of the animal model, appropriate number of animals, selecting exposure routes, duration and levels, and the selection of endpoints to evaluate, and also the building of the exposure system and quantifying exposure during the study, which we went into great detail yesterday. I'm going to focus on these top 3 bullets. This is an exhaustive list. This is just hitting some of the highlights with the designs. So the NTP uses 2 rodent species to evaluate chronic toxicity and carcinogenic activity. This is common with the 2-year bioassay. We use the HSD Sprague-Dawley SD rat. Over the years, we have moved away from the Fisher 344N. This was based on a workshop that was held in 2005. And we have settled on the HSD Sprague-Dawley rat. And this was based on acceptable levels of fecundity, which I'll get into in a minute with perinatal exposure, acceptable levels for body size, and background rates of tumors. The other species that we use is the mouse, and this is the B6C3F1N C6N mouse. This is a cross between the C57 black and the C3H males. We've used this for many years. This was identified many years ago by NCI after evaluation of inbred mouse strains, and it was found to have a hardy lifespan and acceptable level of background rate of tumors. Typically, we use 50 animals per dose per sex for the evaluation of these long-term exposures. However, due to the concern of low incidence tumors, 90 animals were used per group in this study. So as we talked about yesterday, our exposure language is a little bit different with these RFR studies. Instead of chemicals, we're talking about waves of electromagnetic energy. Instead of concentrations, we're used to milligrams per kilogram or parts per million. We're talking about specific absorption rates, power, and field strengths. Instead of material reactivity or stability, we're talking about interference of signal, absorption of energy, and heat of materials or metals. And with frequencies and modulations, this is kind of like enantiomers. And then we spent a lot of time yesterday talking about the exposure system. This was a whole body exposure, but due to the uniqueness of generating a signal and measuring and quantifying that signal and absorption rate, this is a little bit different compared to our feed studies and gavage studies. So the route of exposure is typically based on human exposure patterns. This is, for example, dietary exposure, occupational exposure, whole body exposure. And the duration of exposure is often daily throughout a 2-year time frame. With widespread exposure of particular agents leads to concern that we have exposure during gestational and lactational periods of development. And so the NTP several years ago has started including this perinatal exposure in the rat assessment. This includes the time periods of gestation and lactation. And from there, we usually wean a couple of pups from each litter and continue exposure. And for the particular RFR exposure, we wean 2 to 3 pups per litter. And the selection of a study's exposure levels is based on balancing two things. It's the tolerance of the animal model to the agent of concern and the desire to challenge the animal model for hazard identification. And to estimate tolerance, frequently we conduct short-term exposure studies by the, to identify this tolerance. And Dr. Wyde talked about the 5 days exposure yesterday, and today we'll be talking about the 28-day exposure. And we use this information to set a maximum exposure level. This is selected, and additional lower levels are spaced accordingly. And for the endpoints, typically biological endpoints include survival, body weight, clinical observations, gross and histopathologic evaluation of roughly 40 tissues. We often include interim evaluations to identify potentially ongoing toxicity. This can include body and organ weights, clinical pathology, clinical chemistry and hematology, reproductive endpoints such as sperm parameters and vaginal cytology, additional histopathology, genetic toxicology with the micronucleus assay and the COMET assay that were conducted in these studies, which Ms. Christine Witt will be talking about in a moment. But once we gather all this information and collect all the results, we conduct an analysis and interpret the response. And I just want to highlight that we don't rely on one specific item for determining the response. We review the statistics, dose relationship. We look at the commonality of these lesions. We use concurrent and historical control data if available, looking at latency, progression. We also take into account pre-neoplastic lesions, survival, potential body weight effects if that can affect neoplastic response, structure activity correlations, in particular findings from the other sex or species may be supportive. So if we have a strong finding in the female and we see something in the male, we may see some concordance between the sexes. And also combinations of neoplasms when they originate from the same tissue of origin. I'm going to spend a little bit of time talking about the statistics. We made a couple of adjustments during the analysis, and then talk about the concurrent and historical control data that were provided in the reports. So the analysis of the neoplasms and non-neoplastic lesions, we took into account the survival and the presence of littermates. Neoplasms, as we're all familiar with, usually take time to develop, and the survival will affect this incidence. I mean, if you have differential survival, this can affect your response. In addition, we had litter mates where we took 2 to 3 pups per litter, and so we may have a genetic or environmental component that may lead to an intra-litter correlation. So here's an example of just a data table without any adjustment, where we have the sham control, low dose, medium dose, and high dose exposures, the number of animals with the tumor, the total number of animals listed here with N across the groups, and then the proportion of animals with the tumor right here, going across the bottom here. And so without any adjustment, we are testing for the null hypothesis here that there's an equal proportion across the dose groups for the trend test, and then a pairwise test where we're testing, comparing each group to the control and testing against the null hypothesis that they're similar. When we talk about adjustments, we're going to be looking at adjusting these relative numbers of N here. So for quite a while now, we've conducted survival adjusted statistics on the analysis of lesions to account for differential mortality. And so for a given site, each animal is assigned a risk weight. And so if an animal survives the duration of the study, or when it dies and it has a presence of a tumor, the weight is provided as 1, and the total time is T max. However, if an animal dies prior to terminal euthanasia, we assign a relative risk weight, which is a fraction of the study time it survived, raised to the K power, which is typically K equals 3, and we have our poly 3 statistics. And so here is just an example of this, where we have a fraction of the time when the animal didn't survive all the way to the end of the study. We sum these weights and then alter our, or adjust our N value there. With litter adjustment, since we are using litter mates and we can have this intralitter correlation, this could lead to an overstate the effective sample size and lead to a smaller p-value. And we adjust for this by using a Rao-Scott approach, which is to estimate the effective sample size by using a ratio of the variance of the proportion of animals with a tumor considering litter effect, and estimated variance of proportion of animals with a tumor without a litter effect. And we calculate a design effect here, and then we adjust our sample size by dividing by this design effect and getting to an effective sample size. So for an example here, we have the two extremes where we have 25 litters of 2 siblings each, and then we have 50 independent animals, assuming there's absolutely no intralitter correlation. We take the variances between these, and this is a factor by which we reduce the sample size if we had 50 animals in the study, and yield the effective sample size, and then this effective sample size is then used in the poly 3 test. Moving on from the statistical analysis, I want to talk a little bit about the historical control. The NTP believes the concurrent control of a study is considered the most appropriate, and the historical control data, although do provide us some context of response in a current study. And so we use previous NTP studies within a 5-year window to provide some context to the responses that we're seeing in the concurrent study. But it's well known that species, the strain, the stock of the animal, the feed, the route of exposure, the laboratory, all can vary this background for specific neoplasms. And as identified yesterday, the route of exposure for these particular studies was unique, and we do not have any replicative studies within our historical control database. And what we do have available is a database for the HST Sprague-Dawley ST rats is rather limited. We have recently moved to this animal stock, and this database consists of 4 studies, and this is including the concurrent control. And our database of brain neoplasms is even smaller. We have only 3 studies, and this is due to a change in the evaluation of the brain, where we've increased the sections from 3 to 7. A couple years ago, when we provided the interim report, we did do an evaluation of studies that were at the contract lab, just to get a sense of the consistency and the background response for the brain and the heart. However, these studies are not completed and thoroughly reviewed, so they are not included in our NTP technical report that we're reviewing today. For the mice, we have a historical control database of 11 studies. This is primarily made up of inhalation studies and gavage studies, and it also includes the concurrent control. And so, lastly, I want to briefly talk about the hazard identification. So we conduct these studies to identify potential agents that can increase toxicity and carcinogenic activity. The strength of the neoplastic response or the lack of the response is categorized into these levels of evidence. And these levels of evidence are meant to communicate our confidence in interpreting the carcinogenic response of an agent of interest under the specific conditions of the study. So we're only going to be evaluating the elements within these draft technical reports and the studies that we conducted. And so we have 5 categories of levels of evidence of carcinogenic activity. The top 2 are clear and some evidence, which are considered positive responses. A clear evidence is a dose-related increase in malignant neoplasms, an increase in combination of malignant and benign neoplasms, or a marked increase in benign neoplasms if there's an indication from this or other studies that tumors will progress to malignancy. We have some evidence of carcinogenic activity when there's an agent-related increase in incidence of neoplasms, but the strength of the response is then less than what we require for clear. We have equivocal evidence. This is a marginal increase of neoplasms which may or may not be related to the exposure. No evidence, and then inadequate study when there's issues that preclude interpretation of the study. So, in summary, there are several elements that go into or taken into account for designing chronic toxicity and carcinogenicity studies. A variety of factors are used in interpreting the responses. I highlighted 2, but I will say in the discussion, please evaluate all the ones that we take a look at. And this is including your dose response, your combination of neoplasms. And lastly, the strength of the carcinogenic response is reflected in the levels of evidence categories. And so we have those 5 categories which you will be evaluating in our application of these to the responses that we're seeing in the study. And with that, I'll take any questions.
Thank you, Chad. I'll now open this up for questions from the panel. Yes, Dr. Ackerman.
Yep. So I have 3 questions.
Sure.
One on historical control data. Was there historical control data from ITRI where it was conducted?
No. This is the only study that we have available.
Okay. And number 2, you said in your slides for lower doses, for the low and the mid, that additional lower doses are spaced accordingly. According to what?
Well, so we take a couple of factors, and if we want to get a good capture a wide dose range, if we want to do some greater hazard characterization, sometimes we'll shorten the dose range, only do by halves, because we want to capture the toxicity information within a short dose range, so we can make a hazard identification. Also, it depends on the route of exposure. If we have feed studies where we don't want to go too close together because there may be some overlap in dose exposure, so a variety of different factors.
So in this particular study, how was that scientifically determined?
So I think I'll let Mike answer this with that one.
So how did you determine the low and the medium doses? I understand, there's a lot of rationale and experimentally for the high doses yesterday, and that was really nicely presented. But I was wondering, how do you determine the low and mid doses?
All right. Well, we started off with the thermal pilots. We wanted to cover a wide dose response range. So we started, and we wanted to test the upper range. So that's why we went up to 12 watts per kilogram. We had only 3 exposure groups because of the way that the system was designed. So when we got to the 28-day study, we were looking, as I mentioned yesterday, to push the dose as high as we could. And we looked at half doses, third doses. So it was 9 watts per kilogram, 6 watts per kilogram, and 3 watts per kilogram, I believe, so we could cover a wider exposure range. When we got to the 2-year studies, we wanted to top out at 6 watts per kilogram, But we expanded the dose range down to the low end. If we went the way that we did, we could reach the 1.5, which was close to the 1.6 limit. That's the regulatory agency that's regulated by the FCC. Again, we heard yesterday that this isn't the whole body exposure limit, but for that 10 gram or the 1 gram piece of tissue, that's what that exposure limit is. So I think it extended our exposure range up to the high end, which we believe covered the challenge of the animal on the thermal basis and what we were seeing, and the low end for the chronic study extended down to a relevant level that we thought would be appropriate.
Okay. Dr. Bucher, would you like to comment?
Yeah, I think there was also an expectation that we were going to, as we looked at this study, we wanted to increase the power. So we increased the number of animals. We didn't expect to see striking carcinogenic effects. So with a limited power situation, if we would have dropped the dose ranges or the exposure ranges further for the lower levels, the likelihood of picking up something at those lower levels would be diminished. So we kept to the half.
Yeah, thanks. Yeah, that's clear. And then what I struggled with a little bit in terms of the categories was the difference between some and equivocal. And it seems to me of all the categories that difference is really a gray area. And I wonder if you maybe give a little bit more clarity of what's the difference between some and equivocal.
So that is, to put that succinctly, it's essentially the bright line of, do you think the agent is actually having a positive effect? And this is an effect versus, I'm not sure. We're not sure. It could be, it could not be. And so that's, I think, the simplest explanation. If you think it's some evidence, you actually think this is related to the agent's exposure without hesitation, versus equivocal is you're not sure. It may be, it may not be. So that is to capture sort of the low confidence in the response.
To further that question, when you're at that point of decision making between those, how do you is that where you use historical control data in terms of trying to figure out to tip the balance, so to speak?
Historical control data is used, and within these studies, it's rather limited, especially with the rats. We use a variety of factors, and then it's also a group of us that are discussing this, and so we'll have arguments back and forth, where we feel with the confidence, and sometimes it's just with the group discussion, you may have the outcome being essentially we're not sure, and so that will translate into an equivocal call. Versus we believe this is a strong response that indicate that it's positively associated with the chemical or the agent of interest.
Dr. Felter?
Susan Felter. So I have a follow-up question on the historical controls for the rats in particular. So other than the brain, there were 4 studies including this one.
Yes.
So, and that was 90 animals out of a total of 240, right?
Yeah.
So, if you want to consider whether whether the concurrent controls are somehow different than historical controls, you wouldn't want to be considering them all together. I mean, especially since it's overwhelming your underlying database. And clearly there's a challenge here with a limited database, but why not keep the historical controls separated from the concurrent one instead of lumping them together in the analyses?
So that is a fair point, and that was for the history of the NTP, we have been doing that, and that's frequently because we are reviewing several reports at the same time, and so instead of just removing historical controls, we just kind of lump it together. But in this case, the RFR, the 90, does heavily weight the historical control aspect of that, and that's a good point. And we did look at look at them individually and separate to see if there are major differences.
Dr. Shockley, would you like to comment?
Yeah, so Keith Shockley. I just wanted to make the point that the concurrent controls are used in the statistical testing. So we do not use the historical controls as part of the statistical testing. So the concurrent controls are the most important in that regard.
Right, so, and I appreciate that. I did my best to look very carefully at those tables, and in all of the tables there's footnotes referring to the historical controls and what the incidences in the historical controls are, but then it's a little confusing if that, significant fraction of that is actually the concurrent controls. Because then by definition, you're not out of range of the historical controls because you've included the controls from this study, right?
Right. So in addition to the statistical testing, we also look at the range with the historical controls. So that's a separate consideration. Yeah.
And more to that point, if we did, and there are some cases in these studies where we cite the historical control, and then we will point out that in this particular study, this is making up the lowest or the highest that we've seen in our limited database. So try to make sure we capture that.
So I've got several people here, but Dr. Booger, did you want to comment on the historical controls?
Yes. Well, I just wanted to mention that typically in years past, and I don't think we did it in this particular report, we've included in an appendix, we've included each study that goes into the historical control for that particular set of studies. So, and that way you can kind of do the math yourself, and we'll do that in this particular instance as well in the next version.
So of the others who have indicated they wanted to comment, is this related to historical controls, Dr. Kaufman, or a different question? Go ahead, and then Dr. Lin, you'll follow Dr. Kaufman.
I just have a question to the design of the study. The nervous system is one of the, let me say, important aims or goals of what we want to address with this study. I know it's a cancer study, but what kind of elements did you consider when planning the study with respect to neurobehavioral examinations, observations during the study, or how was it reflected at the end in the pathology design and planning?
So Mike can add to this, but due to the constraints of the room, we were not able to do extended clinical observations in the animal room. We did do a pathological evaluation of all the nervous system, and we have increased our sectioning of the brain from 3 to 7 sections to give a better idea of what's going on there. If there's anything else, I might.
No, I would just agree with Chad. We didn't design any neurobehavioral within the study, and then, as you mentioned, the closed chambers didn't allow us for observations during the exposures themselves. I'd let one of the pathologists talk to the pathology issue.
Dr. Lin.
Staying with the control issue, I'm interested in finding out when and how was a decision made for the study itself, not the historical control, to have one common group as a control group?
Actually, Mike, do you have an answer to that?
I think the decision to do the common control was more of an issue, and this preceded my time, but I think it was more of an issue of cost of additional chambers and the space that was involved in adding additional chambers. In this case, we'd have to have added 3 additional chambers. And since what we were looking at was the absence of the RF, we felt that a common control would be appropriate for both of these modulations.
Well, that's a reasonable explanation, except for the fact that the historical control is really not of much help for the study that we are discussing. So that's why I asked how was the decision made to go ahead in that direction, other than the fact that there is exposure chamber limitations.
So I think the other consideration was that we increased the number of animals to almost double, which was another attempt to try to accommodate this issue. But Michael's correct, the additional 3 chambers and large financial investment in the study at that point were taken into consideration.
But still, though, you have, say, 90 to 100 controls versus several hundred exposed.
That's correct. And if we could do this over, I think we would probably have a second control group.
Dr. Harkema?
Sorry, I actually had 4 questions instead of 3, and I forgot about the 4th. I just wanted more clarity on the historical controls of the mice, because I kind of disagree with you in terms of there is one issue where historical controls in the mouse study plays a very important part with the lymphoma. So I want to just be clear on exactly how many studies were included in that one.
So that includes 10 studies, separate studies, and then the additional RFR, so it makes a total of 11 studies. But it's 10 studies, 5 of which were inhalation, 4 were gavage, and then there was 1 feed study.
And you didn't include this study in that?
We did. We treated this, yeah, like the rats, we included the concurrent control in that control database. Yeah.
Okay, I got it.
Yeah.
Thanks.
Yeah, if I may.
Dr. Lin, yes, go ahead.
Well, I think a couple of things that came out during the panel here that was not apparent while reading the reports. One is that the historical control really is of no relevance to the current study because, for example, the lighting was entirely different. And secondly, we have heard today that there's a limited number of historical controls. At most, there's 3 sets for the rats anyways. So there's a lot of disconnect here as far as how much to rely on the historical control to shed light on the study we are reviewing at this point. So for that reason, I think it would have been, like Dr. Booker had indicated, to have included more controls in the study. Of course, we're talking about something that was in the past, so one could wish, but that's something that one could think about going forward.
Any other questions for, yes, Dr. Barnes?
Yeah, just a comment on historical controls. And the assumption, of course, here is that you're looking at a linear system with regard to dose response. And in some sense, your historical controls are not exposure free of the RF radiation we're looking at. The levels are definitely very low, but it's not a linear system in terms of the experiments we've been doing. So I would say I would be cautious in treating controls as unexposed in the past, because you probably did not measure that in the past, and they may or may not wind up actually being controls in the sense of the experiments you're doing.
And that's actually not uncommon for some of our chemical exposures. Sometimes when we have widespread chemical exposure, we'll have a very low concentration in the control, so it's not a completely unexposed animal.
Dr. Harkema, did you have one more question?
No, just one clarification with historical controls and personal bias on this. So I'm not a big fan. You certainly don't use historical controls for your statistical analysis. That's clear. Okay,
Dr.
but it's informative for the outcome. And in the case with the mice, we'll get into that more. The lymphomas are a concern of why they're so low compared to a lot of other studies.
Dr. Rinke.
Yeah, I just wanted to know whether these mice that were taken in Chicago or also the rats were from the same breeder, from the same supplier than the other ones that were used for the historical controls.
To the best of my knowledge, certainly the rats were, they're all Harlan Sprague dollies. I think that what we do for the historical controls is when we have, in the mouse studies, for example, we have a 5-year window that we use for our historical controls. Because there is a little bit of drift in the historical control backgrounds in these tumors, so we restrict it to a 5-year window. My recollection is that we probably were using mice from the same supplier during that entire 5-year period, but I'm not absolutely sure for the first year or so of that 5-year window.
Dr. Felter?
So I want to follow up with maybe some personal bias as well on the historical controls. I agree there are significant limitations and real serious reasons to heavily consider the concurrent controls. But the importance of the historical controls is also that we have a wealth of information on variability in normal spontaneous tumor response for certain tumor types versus others and the genetic background of these animals. And I think that can be very informative, especially if you see something that's highly variable. And some of the tumor incidence data that we're looking at here were variable too. And so I would caution us to not disregard the historical control data.
Thank you. Okay, so thank you, Chad, for that presentation. We'll now have Ms. Christina Witt to talk about genetic toxicology testing.
Looking for my cursor, there it is, okay. Well, good morning. I'm going to give you a brief overview of the genetic toxicology studies that were conducted as part of these RFR studies. The results of these studies are going to be included later in the course of the meeting in the presentations by Dr. Wyde on the bioassay results. So I'll first present the rationale for selecting the assays that we conducted, and then I'll describe the protocol features of each assay along with the criteria that were used to evaluate the data and interpret the data. And finally, I'll explain how these data aid in developing a comprehensive toxicity profile for a test article. Now, for these gene tox tests, subsets of mice and rats were removed from the exposure chambers after 14 or 19 weeks of exposure, respectively, and assessed for chromosomal and DNA damage using 2 independent assays. The micronucleus assay, which measures structural and numerical chromosome damage in peripheral blood erythrocytes, and the COMET assay, that measures DNA damage in any of a variety of different cell types. Now, in our studies, our COMET studies, we analyzed DNA damage in cells from 5 different tissues: the frontal cortex, hippocampus, cerebellum, liver, and peripheral blood leukocytes. The 3 brain regions were selected in response to concerns that have been raised by some over the years with regards to a possible association between cell phone use and brain cancer. And liver cells and leukocytes were included because these are cell types that are typically included in our other COMET assays. So why these 2 particular gene tox tests? The in vivo rodent micronucleus assay detects alterations in chromosomal number and structure in dividing cells. We all know that chromosome damage is associated with cancer, with birth defects, with some neurodegenerative diseases and other adverse human health effects. Positive results in this assay are well correlated with cancer in rodents. The majority of known human carcinogens that have been tested in the assay are positive in the rodent micronucleus assay. And elevated levels of chromosomal damage are associated with elevated risks for cancer in large human population studies, several of which have been published over the past several years. So there's ample evidence, or there's ample rationale for looking at the ability of a compound or a test article to induce chromosomal damage. However, the micronucleus assay is restricted to measuring events that occur in rapidly dividing proerythrocyte cells in the bone marrow. And therefore, to expand our ability to assess the impact of an exposure on genetic damage in other cell types, the COMET assay serves as a very nice complement. to the micronucleus assay in that we can use that assay to evaluate DNA damage in essentially any cell type of interest because cell division is not required in the COMET assay. For genotoxic carcinogens, DNA damage is a necessary but not a sufficient event because of the multiple fates that damage may undergo. So depending upon the nature and the extent of the damage may be correctly repaired, and we believe that this is the most frequent outcome of the damage that's detected in this assay. Alternatively, a cell that sustains damage may die. And another possibility is that the damage may be repaired but incorrectly. And in that case, a heritable change in the form of a mutation or a chromosomal aberration may be introduced. And I should also mention that these two particular assays are well-studied assays. They've been conducted in a number of labs. They're governed by international guidelines developed by the Organization for Economic and Community Development. So they're very standard assays. Now let's talk about the micronucleus assay. I said that this is dependent on cell division. In the erythrocyte micronucleus assay, pardon me, it's the rapidly dividing erythroblast population in the bone marrow, which is the target. Now, should a hit occur in one of these cells, the resulting broken or lost chromosome will fail to incorporate into the nucleus of either one of the daughter cells produced during cell division. And at the subsequent interphase, that small piece of genomic material forms its own micronucleus, which is very similar in appearance to the main nucleus except for size. Now then, during the process of enucleation, which is unique to erythrocytes, the micronucleus remains behind in the cytoplasm. So therefore, red blood cells that contain micronuclei are very easy to identify, and this is an all or none observation, and therefore the assay lends itself very nicely to automated scoring. In NTP, we use flow cytometry to evaluate blood samples for frequency of micronuclei. There are several advantages to this approach over slide-based scoring methods. Number one, we eliminate subjectivity from the scoring process. Number two, we greatly increase or enhance the power of the assay for detecting chromosomal damage because of the increased number of cells that we can score using flow cytometry. And another distinct advantage is that we can very accurately distinguish between immature and mature erythrocytes in the peripheral blood by using a fluorescence antibody that targets active transferrin receptors that are very plentiful on the cell surface of immature erythrocytes. In mice, in the peripheral blood, we can evaluate micronuclei in both cell populations, immature and mature. And micronucleated mature erythrocytes are in steady state in mouse blood after about 30 days of continuous exposure. Now, the same basic principles of the erythrocyte micronucleus assay apply to rats with one major difference. In rats, only the very youngest fraction of immature erythrocytes can be assessed for frequency of micronuclei because the rat spleen is very efficient at identifying, sequestering, and destroying damaged erythrocytes soon after they enter the peripheral blood. And therefore, this ability to be able to target the immature erythrocytes by virtue of the active transferrin receptors on the cell surface is critical to the success of the assay using rat blood. Because the assay is automated, the scoring is automated, we also evaluate concurrently the mature erythrocyte population, but that is not for rats the population of interest. Mature erythrocytes are, however, used in the calculation of determining percent immature erythrocytes, which is a measure of bone marrow toxicity, and this metric is also generated in the mice. Now, the COMET assay, as I said, detects DNA damage. Breaks can occur endogenously through the normal action of enzymes involved in maintaining DNA integrity, or they can be produced after exposure to a DNA damaging agent. A variety of different damage types may be induced and detected by the COMET assay. Now, the principle of the assay is very simple. DNA is normally in a supercoiled state, so breaks in the DNA will relax the supercoils and allow it to migrate out of the nucleus in the presence of an electrical field, forming an image that resembles a comet. So in practice, tissues are collected at necropsy and disaggregated rapidly to form single-cell suspensions. Cells in this suspension are then combined with agarose and layered onto special COMET assay slides that contain wells. Cells are then lysed to remove cellular and nuclear membranes, leaving only the tightly coiled DNA embedded in the agarose. The slides are then placed into alkaline buffer in an electrophoresis tank, and the DNA is allowed to relax and unwind. An electrical field is then applied, and the negatively charged, relaxed, and fragmented DNA is allowed to migrate out of the nucleus into the direction of the anode, and this forms an image that resembles a comet, hence the name of the assay. Now, following electrophoresis, slides are rinsed, stained, and evaluated using an automated image analysis system. And you see that image here. Let's look at this a little further. The metric that is used to quantify DNA damage is percent tail DNA. And that is, if I can get my cursor working here, that is the amount of DNA or fluorescence intensity that's located here in the tail region of the comet compared with the entire amount of DNA in the cell. Increased migration of DNA from the head into the tail of the comet represents increasing levels of DNA damage or fragmentation, and this is illustrated in the pictures at the bottom of the slide where you see, starting at the left-hand side, my left, yes, essentially no DNA damage in the cell, and then we move to gradually increasing the amount of DNA that's in the tail, representing increased DNA fragmentation. You can see that the size of the head, the fluorescence intensity of the head, is reduced as we get more and more DNA into the tail until we get over here to this image on the very left-hand side of the slide called a hedgehog. That's because to some this resembled an English hedgehog, and hence the name stuck. Hedgehogs, you can see, do not have an easily discernible head, and therefore the software cannot evaluate percent tail DNA in images like this. So hedgehogs are not included in the overall evaluation of DNA damage, but they are tabulated independently. So let's talk a little bit about data analysis. In the micronucleus assay, we analyzed 20,000 immature and approximately 1 million mature erythrocytes for presence of micronuclei. In the COMET assay, 100 cells were scored per tissue per animal. Both sets of data were analyzed using the same types of statistical methods, so that we conducted both trend tests to look at a dose response, as well as pairwise comparisons of each treated group to the sham control to look for significant dose groups. Both tests had significance set at 0.025. And for a test to be judged as positive, we required both a significant trend and a significant dose group, or in the absence of a significant trend, 2 significant dose groups. If only one of the requirements was met, the test was judged to be equivocal, and in the absence of any statistical significance, the test was judged to be negative. Now, in the initial scoring of the rat samples, we noted a marked amount of interanimal variation among animals within a treatment group. In an attempt to try and resolve some of that variation, we went back and we rescored all the rat slides, evaluating 150 cells instead of 100 cells per tissue sample. And this also is consistent with the new OECD guideline that was just published in 2014, which recommends scoring now 150 cells rather than 100 cells per tissue. We were not able to, by scoring more cells, resolve this amount of variation, but even though we saw minimal variation among the mice in a treatment group, we did go back and rescore some of the mouse slides also in order to have some consistent comparative data sets. So how do we interpret the data from gene tox tests? Micronuclei, as I said, are biomarkers of chromosomal damage in dividing cell populations, and chromosome damage is associated with a number of adverse health effects. Importantly, a positive result in the rodent micronucleus test is well correlated with cancer in rodents as well as in large human population studies. A negative result in the micronucleus test, however, does not predict a negative result in a cancer test. Now, positive results in a COMET assay are also concerning. However, if you recall that DNA damage is not sufficient for an adverse event, that damage has to be converted into a heritable change that can be passed from cell generation to cell generation. And there are a number of factors that can influence the fate of this early DNA damage that's detected in the COMET assay. For example, ADME factors. Now, they may be more relevant to chemical exposures than radiation, I don't know. But that is one factor. The type of DNA damage that's induced is an important factor to consider, as well as tissue and species-specific DNA repair processes. So in conclusion, exposure-induced genetic damage is an adverse effect. I think we can all agree on that. But the health consequences of the induced damage are not necessarily predictable. Conversely, lack of genotoxicity as detected in standard assays does not necessarily imply that exposure to an agent carries no risk for adverse health consequences. I think you all know there are plenty of non-genotoxic carcinogens. So genotoxicity is one of the 10 characteristics of carcinogens. It's a critical component of a comprehensive toxicological profile of a substance, but the consequences of the observed genotoxicity in terms of specific human health effects cannot be easily predicted because there are many factors that determine the eventual outcome of this damage. And with that, I will be happy to answer any questions.
Thank you. Questions from the panel? Yes, go ahead.
Mark Klein, Wake Forest. For the COMET assays, you did COMET assays in brain tissue?
Yes, we did. 3 brain tissues.
How did you select cell types to do the assay?
Only a small piece of tissue is taken and a single cell suspension is created from that. So there isn't like a microscopic selection of particular cell types in that we just looked at sections of the brain, the hippocampus, cerebellum, and frontal cortex.
Okay, so they might have been neurons, and they might have been glial cells, or another cell type.
That's correct.
It could have, anything was fair game.
That's correct.
Okay, and then just to comment on the micronucleus assay, there are several tests that are called the micronucleus assay that are different, like the cytokinesis block, ex vivo.
Yes, that's an in vitro test.
Right. So I would just, in the report or in speaking about the test, it's probably wise to call it the erythrocyte micronucleus test or something like that to avoid confusion.
Thank you.
Dr. Harcum?
So in the COMET assay, when you take it from several different sites in the brain, and first of all, how do you determine what sites you want to look at, number one, and number two, how is that statistically handled as individual? Do you lump them together or do you keep them separate? And if you do that, is there a certain weight put on one versus another?
Each of the tissue types are considered independently. They are not combined. The selection, frontal cortex was selected because that apparently was of interest in terms of the possibility of brain tumors, and hippocampus and cerebellum were just selected because they're other fairly good-sized portions of the brain, and we were just trying to cover our bases, essentially. John, was there any other? Yeah.
And so I know that the pathology group has gone to several more levels of looking at the brain than conventionally, and was that taken into consideration? I'm not sure how many levels were actually in this study, 7 or something like that. And so my question is, was it the COMET assay a good representation of the whole brain? And maybe it's a nitpicky a little bit here, but if you're going to compare histopath and a COMET assay, you would like to at least understand or be informed on that. So, was I clear there or not?
Yeah.
Okay.
To follow up, we had 7 sections of brain cut in. That's just determined by the anatomic sites that the 7 sections are taken. But we also looked at spinal cord, 3 areas of spinal cord: cervical, thoracic, and lumbar, and peripheral nerves, including trigeminal nerve and the femur, the femur. So, but those are all looked at histologically, and there was no correlation or finding that correlated with the gene tox.
But just to remember though that the animals that were taken for the COMET assay were not part of the 14-week interim sacrifice that were done for histopathology. Just
Dr. Adler.
Rick Adler. So question back to the COMET assay, and maybe you went over this and I missed it. Is there a threshold for positive versus negative in the software? Where is that line? And is there a positive control run for that assay? What's the positive control agent used?
In these NTP animal studies, there isn't a positive control per se because we don't have a positive control group in these chambers. But we did run, and this is mentioned in the report in Appendix E that deals with all the gene tox studies. We did run slides that were human TK6 slides that had been exposed to a genotoxic agent that induces DNA damage. And we ran 5 of those slides with each one of the sets of slides in an electrophoresis chamber as sort of an internal technical control for the assay. So, and you had another question.
What's the line in the software for positive versus negative? Is it based on that internal control?
The software doesn't tell us what the baseline is for positive control or for negative control or where we've exceeded that. That's part of the laboratory historical control for the particular assay or the tissue. Each tissue has its own baseline level of damage that's typically seen in the tissue. And this particular study, as you've heard, it is so unique in so many different ways that we do not have historical controls for the COMET assay for this study.
Dr. Lin and then Dr. Pan.
Yeah, in addition to the neural tissues, did you happen to assay tissues from other parts of the animal?
The liver and peripheral blood leukocytes.
Okay. So those were the only ones that you did?
Those are the only other tissues, yes.
Okay.
Dr. Pan?
So, to answer your question, the perceptive instrument scoring system that we all use for COMET assay, if 90% of the DNA is already in the tail, they would consider that as the hedgehog or cloud. And that particular cell will not be used in calculations. However, we keep track of them. So that's the only thing that we have. You were saying that what the software will tell us where the positive is. So that's the only thing.
So I have a question relating to the positive predictive value of the micronucleus, erythrocyte micronucleus test. You said it was very high. NTP must have a lot of studies in which this has been looked at in terms of positive predictive value and negative predictive value. And I'm just curious in terms of the number of what one would determine as a false positive in which the micronucleus test was positive, but the bioassay was negative. You said it's high, but I'm curious about the relative.
Yeah, the last time a systematic review of the micronucleus data, in vivo erythrocyte micronucleus data was compared with a bioassay outcome was done in the year 2000. We did have a lot of studies by that at that point, and at that point, if I recall correctly, it was something on the order of 95 to 98% positive predictivity. Since then, I am not aware offhand of any chemical that induced micronuclei in vivo that was not shown to be carcinogenic in the rodent bioassay. There may be some upcoming studies that would contradict that, but I don't recall offhand any. Do you, John? No. It's a test with low sensitivity because it's very compartmentalized in the bone marrow. The chemical has to get there, or the agent has to get there, or the metabolite has to get there to induce an effect. So its sensitivity is low, but a positive response is pretty meaningful.
And that's true for both rats and mice?
Correct.
Interesting. Thank you. Other questions? Ms. Whip? Okay, thank you very much.
Okay.
So we will now move on to a discussion on the toxicology and carcinogenesis studies pathologic evaluations from Dr. Amy Bricks.
Good morning. Today I'm going to briefly discuss the typical process that is undertaken to review the pathology data from chronic studies undertaken by the National Toxicology Program, with specific reference to the 2-year study of cell phone radiofrequency radiation in mice. I'll first mention the role of the study pathologist in generating the data, then go into a bit of detail concerning the process that is used to assess the quality of the pathology data, including the pathology data review, the audit of pathology specimens, the pathology quality assessment slide review, the pathology working group, and the final steps. The study pathologist, a board-certified veterinary pathologist with experience in toxicologic pathology, supervises the necropsy of and reads all of the tissues from all of the animals in the study. The mouse cell phone RFR study was a big study due to the fact that there were approximately twice as many animals per group as would typically be in a chronic bioassay, and also that there were 2 modulations, GSM and CDMA. The tissues for the 2 modulations together numbered approximately 50,000. The study was set up with a control group that was shared between the 2 modulations, as you've heard, so there was only 1 control female group and 1 control male group. The study pathologist will prepare a pathology report, which is part of the study lab report. The study lab report, slides, blocks, wet tissue, and all associated documentation are shipped from the study lab to the NTP archives, and at this point, a review of the pathology data, or PDR, is done. This review is done by a quality assessment pathologist, or QA pathologist, at one of the NTP pathologist contract laboratories. QA pathologists have no relationship or communication with the study lab or the study pathologist. However, they are in close contact with the NTP study pathologist, which in this case for the mice was me. My job as the NTP study pathologist is to oversee the pathology review process and to make sure that the final data are accurate and consistent both within the study and with other studies that the NTP has done. The QA pathologist and I will review the study lab report, as well as the study lab report for the companion study, the statistical tables, and the summary tables. We compare the incidences in these studies to those observed historically in B6C3F1N mice, and then the QA pathologist prepares a draft PDR report. The draft PDR report includes recommendations for which tissues will be reviewed during the pathology quality assessment slide review. It includes organs that will be reviewed for all lesions and organs that will be reviewed for only specified lesions. It also includes other lesions that will only be reviewed when they were diagnosed by the study pathologist. This last category is done mainly for terminology changes. Terminology changes made up most of the changes to the data in the mouse cell phone RFR study, and they were done because between the time the study pathologist read the study and the pathology data review was performed, the NTP had published their Atlas of Non-Neoplastic Lesions online, listing their preferred terminology. The PDR culminates in a data review meeting involving the QA pathologist and pathologist, toxicologist, and the statistician from the NTP that are involved with the study. At this meeting, a final decision is made as to which organs and lesions will be reviewed during the slide review. For the mouse cell phone RFR studies, it was also decided to do a complete review of all of the tissues from 10% of the control and high-dose animals to make sure that no lesions had been missed. This complete review is typically done in short-term studies, but less commonly done in chronic studies. Subsequent to the PDR meeting, and before the quality assessment slide review begins, an audit of pathology specimens, or APS, is performed by the QA pathologist. Residual wet tissue is examined to make sure all potential gross lesions have been trimmed in, that all organs have been properly incised or opened, and that animal identifiers, usually tail tattoos, match the identification on the wet tissue bags and on the necropsy records. Slides and blocks are examined to make sure they are present and accounted for, to check for complete and accurate labeling, and to check for proper sectioning of blocks and proper preparation of slides. As is typical in chronic studies, wet tissue slides and blocks from a random 10% selection of the animals in each group was examined during the APS of the mouse cell phone RFR studies. After the audit of pathology specimens, the slide review is done by the QA pathologist. Due to the large number of slides, the slide review of the mouse cell phone RFR study was performed by 2 pathologists, one doing the males from both modulations and one doing the females from both modulations. The slides were divided in this manner due to the shared control groups. Both pathologists worked for the same contract lab in the same location and were in regular communication about diagnostic terms and criteria. The QA pathologist, like the study pathologist, reads the slides in an open or non-blinded manner. The practice of reading a study in an open manner is based on the fact that interpreting tissue changes is complex and subjective and relies heavily on the concurrent controls for identification of subtle lesions. In blinded studies, only lesions that are clearly outside of any normal range can be recorded, so reading a study in a blinded fashion actually decreases the sensitivity of the study. Because of these reasons and others, reading a study in an open manner is considered preferable and is the industry standard. Comparison to the concurrent control group is the most scientifically appropriate and relevant comparison for toxicity testing in animals. It is critical that the pathologist have access to all information about the animal, including clinical observations, gross lesions, organ weights, and dose group when reading a study. Any differences of opinions that occurred between the study pathologist and the QA pathologist are shown to the NTP study pathologist, who resolves the differences. In a few cases, when a diagnosis can't be agreed upon, the slide is set aside to take to the pathology working group for a resolution. After the QA pathologist has reviewed all the slides and the NTP pathologist has addressed all the differences of opinions, the QA pathologist prepares a quality assessment report, and the slides are chosen for the PWG. The pathology working group, or PWG, consists of the QA pathologist, the NTP study pathologist, several other NTP pathologists, and outside experts. During the PWG, the participating pathologists carry out a blinded review of slides with representative examples of potentially treatment-related lesions, as well as lesions that were recommended by the QA pathologist to be added or removed. Photographs are shown to the PWG participants in a PowerPoint presentation for cases where terminology is to be changed, but no substantive change in the lesion diagnosis are proposed. The PWG for the mouse cell phone RFR study was held over 2 days, 1 day for lesions in the female mice and 1 day for lesions in male mice. Subsequent to the PWG, the pathology data is updated, then audited by a contractor who is not affiliated with the study in any way prior to the audit, and finally, statistical and incidence tables are produced. These tables are then used in the generation of the technical report. The process used for pathology review of the mouse GSM and CDMA studies was a similar process to that done for most of the chronic studies performed for the NTP. As with other NTP studies, the slides and blocks are available upon request for review in the NTP archives. Are there any questions?
Thank you. I'll open it to the panel now for questions. Dr. Arkham.
First, just like make a comment. No place does this better than you here at, and that should be recognized. So you're kind of the gold standard. With that in mind, also, I wondered if I could give you an example, looking at the data as a reviewer. Could you bring us through the whole process to ensure that this lesion was adequately looked at? And I'll give you one here, and that is the lymphoma. Okay, so could you kind of, from the study pathologist all the way along the track, to ensure this rather low incidence of lymphoma was verified? And so that's one. And I'm not even quite sure. I'm a little confused too about where is the study pathologist, and usually it's at the place where the where this is being conducted. Was there a study pathologist located at ITRI?
So ITRI doesn't have a study pathologist, so they had a subcontractor, and that subcontractor was locally present, but not physically at ITRI.
Okay, all right. But anyway, first question.
Okay, first question. So specifically to deal with the lymphomas, the study pathologist actually noted that in the report, and we would read the study pathologist report for any conclusions that they might make, because they would also get included in the pathology data review if they had seen something they thought or something. Then in the PDR, we would also look at the statistical tables, and anything that was statistically important, we would add that to the review. So it would also have gotten flagged that way. And of course, the incidence tables we would also have flagged, and we look to see anything that's unusually high or low in the control things compared to historical. So it would have also flagged then. So then we would have reviewed all of the lymphomas that have been diagnosed. As you know, in mice, they get a lot of lymphoid hyperplasia. So sometimes it's just a difference of opinion whether it's hyperplasia or lymphoma. So they would have all been reviewed. All neoplasms in all animals are automatically reviewed. So all of the tissues with lymphoma would have also been reviewed. So the conclusion did not change throughout the study. And then we would have taken representative examples of that through to the PWG. So
then the only other, a little bit different, maybe this is done more routinely than I know because I don't read so many slides like this, but to have two pathologists, one doing females and the other one doing males. And I understand the amount of work and all that, but it seems to me a little bit unusual.
It was unusual, and it was just the size of the project, that we did that. But the two pathologists that read that, like I said, they were in constant communication with each other and both with me. So it was almost as if the three of us were working together.
Other questions? Dr. Lin?
For clarification, would you say something regarding the protocol or the logistics involved, both at the study pathology level as well as review pathology level, how blinding was taking place in each one of those situations?
Okay, at the study lab, they are not blinded at all, nor during the QA review. We don't blind them because it's just considered not as sensitive of a read if you read something blind. If you read something blind and you see something in an animal, you can't record that unless it's outside of whatever normal range you would possibly have. Whereas if you're reading it blinded, you can be much more, sensitive to those findings and subtle findings. Plus, it's imperative if you're reading a study that you know about clinical observations, gross lesions, organ weight changes, things like this. By the time we get to the PWG participants do not know what dose group it is or what the original study pathologist or QA pathologist, what specific thing they called it. So, but in order to be the most sensitive we can with these lesions, and the concurrent control is always the best, so they read it against the current control.
So at the study pathology level, there was no differentiation between knowing whether it's the control animals or whether the tissue came from the control animals or from the exposed animals.
They would have known that, yes. And they would have known like I said, they would have had the necropsy sheet there, so they would have known what gross lesions were there. This is the standard procedure for, all commercial and academic reads. This is the industry standard for toxicity and carcinogenicity studies of how a study is read. It is the most scientifically appropriate method.
Well, I understand your emphasis on sensitivity, but a lot of times in a scientific study situation whereby people go the extra step to make sure that the pathologists were not informed of the source of the specimen.
Yeah, the question we're not really asking in these studies is it or isn't it, but what is different than the controls? And so we need to see all of that spectrum. And it's a biological system, so there's going to be variability. I know there's going to be discussion today about historical controls, concurrent controls. So you have to see the total of the concurrent controls and that biological system that occurred in order to see if something is different than that. You can't just say, absolutely, this is different from an absolute. If you do that, you're only going to be able to record the most extreme changes in that biological system.
I understand that that's one way to look at it. On the other hand, the human beings are always prone to biases of one time or therefore for whatever reason, maybe there are shortcomings and so forth like that. I think that's one reason the blinding process was instituted.
Well, and that's why we carry through the review and ultimately the PWG as well.
And the PWG is blinded.
They're read in a blind.
I understand that. I'm talking about the study pathology.
Right.
And your sense is that that was perfect, like you said, that was a gold standard. So anyone who does not apply that gold standard potentially could be labeled as a non-scientific study.
I don't agree with that, and I think this is the way most studies are read, and most of the information is properly gleaned from them.
So how then the blinding process got introduced into the assay of various specimens?
So if you're a study pathologist and you see something that is very subtle, oftentimes you will go back and pull all the bone marrows and just look at them blinded to see if the thing that you think is hyperplasia is really there, and you will do that. But
so you already know at that point where did that sample came from?
You would just when you read the study, if you are not sure of an effect, you'll take all the bone marrow slides, mix them up, and read them blinded yourself. You will do these things. I mean, the study pathologists, this is the way the studies are read. There is some blinding in reviews and things, but you cannot get an appropriate read if you do not have all that information, because it is a biologically complex system, and those things are all interrelated.
I'm fully aware of the biological vigorous that's involved compared to especially physical sciences. But still, this is an issue that's been brought up by many different people some people have made a big deal about it as well. I'm not sure your discussion here allays those concerns.
Well, I'd like to ask Dr. Bucher to comment on this.
So I would just say that this is not an argument that's unique to the National Toxicology Program. This is a discussion that has gone on among pathologists for a long time, especially those who have to read studies of this type. So, and I think it's an argument that continues, and I think that the Society of Toxicologic Pathology has taken this up recently again to look at it, and there have been publications on this, many publications over the years. So it's, I recognize exactly what you're saying. I've listened to some of the criticisms of this that Dr. Lin's bringing up, and I think it's a very important subject, and that's one of the reasons that we wanted to explain in full the reasoning that went into the way this study was done, as are all NTP studies done in this manner. So it's out there. People can look at it. You can make up your own mind.
So several of our committee members are certified pathologists. Does anybody else care to comment on this issue, Dr. Harkema?
Yeah. So even the pathologists don't take this lightly. Okay, and that's why there was rigorous review of that whole process. And the peer review, I think, as Dr. Bricks stated, the peer review that is the most critical and unbiased is placed in the right level that she's talking about, and from the other studies. And I'll talk, industrial toxicologists here can comment too, but for the state of the science or art of this right now, that bias is handled at the peer review level and will be caught if there's anything there. But having read, and in my former life, having read thousands, tens of thousands of slides on studies like this, if you don't know the controls, the background lesions, everything else that's taken into account, you will you just won't be able to make an adequate, in an efficient, timely manner. In your bias, I do a lot of morphometry as well, which is an unbiased approach to eliminate that. But at the end of the day, if you can't see it, you're not going to count it, and these match up almost always. The morphometry always just validates, put a quantitation onto what the study pathologist has only seen. So the bottom line I'm saying is that the bias is caught, if there is any, and with a multiple number of peer reviews. And then secondly, lots of the morphometry has already validated in 99% of the time, the quantitative unbiased assessment using quantitative analysis rather than semi-quantitative almost always validates the pathologist's outcome.
So in a sense, what you're saying that the review pathologists are more reliable than the study pathologists if there is any discrepancy.
I'm not talking about reliability. I'm talking about where the peer review process is unbiased, I think, is in the most appropriate way. I don't think the study pathologists, I think a lot of times if there is any bias there, it's going to be caught later on. But study pathologists are very, very well trained and don't institute as much bias as you would think.
Dr. Rinke?
Maybe it is useful to add as well that we, over the last 20 years, really tried to get diagnostic criteria that we really have to differentiate from pre-neoplastic to neoplastic lesions. And so the basis of the knowledge of the finding that you have, either blinded or not blinded, is more or less the same. So the final decision then has to be made. And this is frequently these discussions, is this still a benign or is this already a malignant tumor? But the tumor itself first is there, as well as the hyperplastic lesions. And we saw that in the introductory talk that preneoplastic lesions and others were also involved and of course considered to our results here.
Thank you. Any other comments, questions from the panel to Dr. Brooks? Dr. Klein.
Yes, Mark Klein, Wake Forest. Generally, how was the head assessed aside from the brain? Will we hear about sectioning levels at some point, or is this the time to ask that?
You can ask about it. I mean, you take take 3 sections of the head at specified portions of the bone after they're decalcified.
Okay, so one of those is through the ear.
I don't believe we look through the inner ear. Is that what you're asking?
Right. I'm talking about the vestibular system and the auditory nerve and all that.
No, not that I know. Dave.
It wasn't
Dr.
routine,
Malarkey?
but in the rat, we did go back and do some exploration, but not in the mouse.
Okay. Well, thank you. Thank you, Dr. Brooks. We will take a 20-minute break. We're running a little ahead of schedule. I'm sorry, Dr. Felter.
Are we allowed to go backward and ask a follow-up question from the gene tox discussion?
Sure, sure. We will get back to that.
So my question is about how the results from the COMET assay are reported, and you described the positive and equivocal and negative. What you didn't say anything about is what do we do in the case where there is the data support a trend in the opposite direction? So, and the reason it's important is because in the females for both modulations in the frontal cortex, there's actually a statistically significant decrease in the COMET tail, and that's where we saw the positive results in the same assay in the males, right? So if you can help me understand how do we even think about that, since it's listed as negative, but there was a statistically significant trend in the data.
We don't do what is it called? Two-way? What is two-sided? Thank you. We don't do two-sided trend tests, so we can't actually statistically say that that was a significant decrease. It may look like a significant decrease, but we didn't analyze it per se. And the tests aren't designed to detect decreases in DNA damage that might be attributable to cross-linking or something like that. With a high background, you can see a decrease in DNA damage that would make you wonder whether or not you do have a cross-linking phenomenon occurring in that tissue. That would be something that should be followed up then a little bit more rigorously, and maybe do a test where you actually introduce a genotoxic compound, one that's known to increase DNA damage, and then you do your exposure of interest to see whether or not that damage level then drops with increasing dose of your test article. That would be a definitive way to look at cross-linking. Okay.
Thank you. Okay, we are adjourned and we'll resume at 10:20. 20-minute break. Thank you. A brief explanation of the charge to the committee from Dr. Blightstone. Chad?
Okay. This will be similar to the information that was provided yesterday, but as you know, we conduct rodent toxicity studies on agents of public health concern, and we describe these results in the NTP reports, which you will be reviewing today. There's the 5 levels of evidence of carcinogenic activity, which I described in my earlier talk, where the top 2 calls are considered positive. We have an equivocal, which may or may not be related to exposure, and then no evidence, and then inadequate study. These calls are presented in our reports, which all of you will be reviewing. The 2 reports, now we're moving on to the toxicity and carcinogenicity aspects, the biological response to the exposure. The first report will be the mouse report that we'll be reviewing, the exposure to 1900 megahertz with the GSM and CDMA modulations, and then following that we'll have the rat report with the exposure to 900 megahertz with the GSM and CDMA modulations. So the charge of the panel today will be to review and evaluate the scientific and technical elements of the study and its presentation, and determine whether the study's experimental design, conduct, and findings support the NTP's conclusions regarding the carcinogenic activity and toxicity of the test agent. And with that, I'll answer any questions regarding the charge.
Thank you. Any questions from the panel on our charge? Okay, thank you, Chad. So we are now going to enter into our second public comment period. Could I have the next slide? He's getting it up there now. Thank you. So I again would like to acknowledge and thank all of those people who have submitted written comments. And it's important to recognize for those who have provided written comments that those comments have in fact been provided to the panel for their incorporation and their consideration in their own comments. So again, here is the list of those people who have provided us with written comments, and I thank them for their input, and trust that the committee has taken a look at all of these and incorporated the comments into their own reviews. Okay, thank you. So we will begin now with our oral presentation. We have, is Dr. On the phone? We have one presenter from South Korea, I believe, who is going to do this on phone. He's not on phone yet. We're on a little ahead of schedule, so he probably will join us later. So we will put him later in the schedule of oral presenters. So we will now move to Theodora Scarato. Dr. Scarato, we heard from her yesterday, and she'll now be talking about the biological aspect.
Sorry, I'm trying to get the cursor up.
Off screen.
Oh, I think it's okay.
Yeah, there you go. It's just when you move them.
And again, for those presenting public speakers, the same rules as yesterday apply. A 5-minute session. If you could please limit your presentation to 5 minutes, and the timer is there on the podium for you to see.
And if you're speaking on anyone's behalf, please state that also. Thank you.
Okay. People have no idea that we're researching. Wait, do I know? Wait, hold on. I think I pressed the button and it didn't do anything, so maybe I'm pressing the wrong button. Wait. It's not pressing.
Right page up, page down, the arrow.
No. Oh, but
Sometimes the arrows here, page down and page up. You see, sometimes those will.
right, I just want to know what button to press to go to the. Okay, it's working. Okay. Okay, I'm good. Okay. Theodora Scarato. So I think, again, I wish that the public were more aware that we are currently studying an agent that is widespread over the world's population. I'd like to point to a couple comments in the NTP report. The technical report refers to FCC human exposure limits as safety guidelines, but this is inaccurate, as the FCC has adopted maximum permissible levels, not safety limits, because proper safety testing was not ever done on chronic low-level exposure, which is why this very study is being reviewed today. The NTP found adverse health effects, namely cancer at levels the U.S. government, at levels that the U.S. government assumes would cause no harm based on how they set these exposure limits. And the finding of increased cancers and precancerous lesions, I think many scientists are stating, confirms that the basis for our FCC limits are non-protective. I wanted to point out that I think it would be important for the technical report to include the regulatory limits of other countries and briefly summarize that the United States allowable FCC limits are far higher than many other countries' limits. Another thing that is important to add to the technical report is the issue of co-exposures. When we're prescribed medication, the doctor checks for synergies, and you get a side effect sheet that's a mile long, but with electromagnetic fields. With no such safety testing done, nor monitoring for side effects, we don't have that. And there is substantial evidence of potentiation of effects. So, for example, I think that the technical report, I'm sorry, I'm having trouble with this, needs to review studies that have showed synergies and co-exposure. So this slide, I have two studies, Björn 2013, which found increased ADHD symptoms associated with mobile phone use for children with higher lead levels. And the researchers hypothesized this was due to the combined toxic effect of RF and lead on the developing brain. There was another study analyzing over 1,000 mother-child pairs, which also found a significantly increased risk of having poor or delayed neurodevelopment up to 3 years old in association with maternal mobile phone use during pregnancy. There's actually a rich literature on synergies that needs to be summarized in the technical report, and certainly the research showing a tumor promotion effect, both in human and animals, and I don't have time to review all of that. But I'll give you the references. I'm not going to go over all of this, but there are a few additional studies that show permeability in the blood-brain barrier that need to be added to the report. These images are from a study, one of several published studies by a research team in Turkey that found rats exposed to cell phone radiation had decreased brain cells. And in this study, pregnant rats were exposed to 900 megahertz for 60 minutes a day for full gestation and then sacrificed at 4 weeks old and found decreased hippocampal cells. Other studies have found prenatal and postnatal exposure leading to pyramidal cell loss in the hippocampus, decreases in the Purkinje, I say that wrong, cell numbers in the cerebellum. And this research also needs to be added to the report. We know there is no safe level of lead because it damages the brain. And whether in alone or in combination with other exposures as a tumor promoter, the fact that numerous research studies have found damage in the brain, I believe, is critically important to trigger government action. There's also research such as mice exposed prenatally to cell phones in a Yale study, which found increased hyperactivity, poor memory, and altered brain development. Additional published studies have found prenatal exposure linked to behavior problems and speech problems that need to be summarized in the technical report. I also think that it needs to include the worldwide action at kind of local levels on reducing RF exposure to the public. We have one of the most comprehensive databases, Environmental Health Trust, on these policies, which includes, I'm not going to go over it, but all of it, but decreasing Wi-Fi in schools, banning the sale of cell phones for children, and most recently, French Polynesia actually initiated a major public information campaign both on ELF and on RF exposures. In Maryland, the 19-member Advisory Council on Children and Environmental Health and Protection recommends limiting exposures as much as feasibly practical in schools and directs the Maryland State Department of Education to consider using wired devices in classrooms. Most recently, the California Department of Health issued guidelines to how to reduce radio frequency radiation, but what I think is most important is what the California Department of Health actually deleted over 10 years of rewrites, almost 10 years. They deleted recommendations on cordless phones because, of course, the cordless phone base emits radio frequency nonstop, and also is important that these original recommendations were for California state employees. The first draft was information for state employees to reduce exposure and recommended the Department of General Services action to reduce, to purchase reduced devices that have reduced exposures. Currently, 10 years old is the average age for a child to get a cell phone, as if it was some rite of passage. Yet a decade ago, California was clear, do not allow children to use a cell phone except for emergencies. That was deleted, that very clear advice. And I think that occupational exposure, not just for cell tower workers, electrical workers on rooftops, which is critically important and represents an egregious lack of protection, but also for delivery drivers, restaurant, retail workers, state employees, and I would add that the way Wi-Fi is rolled out into schools, students are in the same category. I also noticed that the DNA damage analysis is missing from the NTP technical report as was presented in September 2016. This is the poster from the September meeting, which states that the results suggest that exposure to RFR has the potential to induce measurable DNA damage under certain exposure conditions. However, the 2018 draft has omitted such a characterization, and I think that needs to be added. I also noted that the information on the IR conclusions, that it supports the WHO IR conclusions. That sentence has disappeared in the 2018 report. At least that and reference needs to be added back on. So we have increased schwannoma in the two of the largest studies ever done with radiofrequency radiation. And yes, the Ramazzini study is a different exposure. However, they are concordant with the same agent. And then to have increased risk in humans of the same type of histopath. Case control studies of people have found people who hold cell phones to their head in the long term are at increased risk of acoustic neuroma. I also, and didn't have time to go over these, but there's increased schwannomas were also found in another Ramazzini study that combined ELF, non-ionizing radiation, with gamma radiation. And when it comes to the mice, we have the NTP mice with increased lymphoma in parallel with the Lurchel study, which needs to be included in the technical report, which found increased lung and liver cancers as well as increased lymphoma at levels far, far lower than the NTP. A woman contacted me recently who had a stage 3 pleomorphic sarcoma tumor on her right thigh, directly underneath where she favored placing the iPad for work. We have, and I can't work this, we have young women who have case reports of women with breast cancer directly underneath where the phone was stored in their bra. Is this by chance? So in plain terms, we have more than one study of finding rats getting schwannomas and mice getting lymphomas. And I think that the concordance of these observations cannot be ignored. Considering we're living in a world of multiple exposures, even if radiofrequency were just a tumor promoter, with billions of cell phone users, I think the US government needs to urgently act to limit public exposures. And the public, again, is completely unaware that we have a product which has been untested for long-term safety. And I thank you for reviewing this issue today.
Thank you. Any questions from the panel for Dr. Scarney? Okay, thank you very much. We will continue now with Dr. Olga Naidenko.
Good morning. For the record, my name is Olga Naidenko. I am a senior science advisor with Environmental Working Group, a nonprofit organization based in Washington, D.C. It is a pleasure and a privilege to be here talking to you on the second day of this important meeting. And of course, the EWG written comments have been submitted for the record and should be available now for all public and this important review panel. Moving on to what I would like to address today, this panel has read the news. We all are familiar with the discussion and the importance attached to the issue of safety of all the wireless devices which are around us today, especially the effects on children. After Theodora, that's a tough act to follow. There's not much more I can add about how public really needs to know the right and reliable information and look into the federal government scientists to come up with this kind of information. Rather than focusing on those topics, I would like to talk about something completely different, and that is how can this stuff really work? Because what this panel is reviewing is, of course, the NTP study itself, as Dr. Booker reminded us. But the fundamental framework is, if indeed, as we heard from FDA yesterday, the only thing that EMF radiation can do is hit the tissues, then one would say everything is not real, right? Heat cannot cause cancer, people would say. But again, the NTP is in a unique position to actually move ahead and start digging into the biology. And what I would like to mention is something that people around this table might not know. And that is that there is a company out there, happens to be called NovaCure. I have not met those people, so as federal government scientists say, this is just for information purposes only, not for promoting a specific company. And what this company NovaCure does is that they are now in stage 3 FDA-approved treatment for technologies they call tumor treating fields, which FDA has now allowed for phase 3 as a potential treatment for glioblastoma patients. They are now trying to apply it to a particular type of lung cancer. And the idea there is that the electric fields are applied to the heads of the patients with a brain tumor, supposedly decreasing the brain tumor growth. Now, one of the two things is right. Either that company is pulling the wool over the minds of everybody, possibly over the minds of FDA, although there is a very competent team of people at FDA reviewing drug applications. Many people around this table are very familiar with the very rigorous FDA drug approval process. So it is possible that that company has succeeded in hoodwinking everybody. It is also possible that the stuff actually works. And it is possible that electromagnetic fields not only have biological effects, but those effects are so subtle and potentially could be controlled in ways that not just cause things like affect how DNA divides and potentially affect the DNA stability, but also affect biology in a way that can be harnessed so as to provide cure. My favorite example for this is chemotherapy. No, I'm not saying that cell phones are like chemotherapy. This is just an analogy because analogies can work well. That is, chemotherapy saves lives. But nobody would want to put a sprinkle of chemotherapy on their kids' cereal in the morning every day, and then also on lunch and dinner. So the reason I'm taking you on this little story about NovaCure and Optune, you can Google tumor treating fields, you will see how far in the FDA approval they have gone, is as this panel and as NTP is considering this incredibly complex biology of how EMF can impinge on biological tissues. I suggest that somebody picks up the phone and talks to the FDA people who have approved their technology up to stage 3 and say, does this stuff really works? Because that's what we need to do, is to bring together all people, not just at NTP, but all people who have ever researched EMF and its effects on biology. And it's not just about the last readout in a 2-year biological study. Or for that matter, breast cancer people appearing in women who have their cell phone in their bras. It is very hard to trace that causality, we all know that. It will not get easier next year, and it probably will not get easier in 10 years. But sort of stepping back and looking at the biology, if in fact the EMF fields can affect the cells. That's something that we want to know, because maybe just like chemotherapy, we can find a way to use that to save lives. So, and now of course I need to finish with the official comments which I have submitted for the records. Environmental Working Group does believe that the results of that we already have in hands are showing a risk, especially for children. Translating data from animal studies to people, anybody who has ever done that has been pulling their hair out, because sometimes animals show greater sensitivity to specific agents than people, other times they show less sensitivity. And yet at other times, like we recently found for pesticide chlorpyrifos, it is just impossible to design a kind of a subtle animal study that will show those alterations in brain and memory that we see in pesticide-exposed children, because we don't know what rats are thinking about. Maybe they're thinking bad thoughts and trying to escape from the cage. So I will pause here. I will thank you for the opportunity to comment and repeat the Environmental Working Group position that we think these studies are really important and their application to human health needs to be taken seriously. Thank you for your time.
Thank you. Any questions from the panel? If not, thank you.
Next is
Dr. Deborah Davis.
Good morning. I'm speaking to you today on behalf of some of my colleagues at the Hebrew University who share with me these concerns, and as someone who once sat where you're all sitting as a member of the Scientific Advisory Board for the NTP several years ago, I want to comment on the fact that this is an unprecedented review you are part of. I don't know of a single other compound in the history of this program that has been subject to a 3-day review. And I don't think it's because the science is debatable. I want to congratulate the team members here on doing an excellent job, particularly in explaining to those who may not be familiar with it how blinded pathology does work and why we proceed in the way that we do. I want to add that the comments provided about the international standards are noteworthy. I would point out that the home of the World Health Organization, the home of arguably some of the best technology ever developed here, Switzerland, is also this place with the lowest standards in the world today. This is a very important study that you've done, but I also would add it's not a lifetime study. It's a study that ends at 2 years. And what we know about lifetime, and I see many of us are now well beyond our 60s or looking forward to moving forward, is that in fact 60% of all cancer in humans occurs after the age of 60, and your animal studies effectively end at the equivalent of a rodent age of 60. So as a consequence, we have to take very seriously what you have produced here. Now, I'm going to talk about the fact that it's really important that you stick with the controls in this study and not historical controls because of the unique housing, again, developed by talented engineers, and that the baseline rate of cardiac schwannoma, even in historical controls, is quite low, as your own data suggest from Greg Dinsey, who was one of my co-authors many years ago. And I think you need to re-examine some of the endpoints on reproductive and birth weight, because of course the birth weight impact is not just an impact on the pups, but I suggest it was interfering with lactation, of course, in the dams, which is why you have the birth weight, the continuation problem of low weight when the animals are still in nursing. Now, we see DNA damage, a multi-site and multi-site increased cancers. While they may not be statistically significant, I think sometimes we confuse statistical significance and biological importance. And I believe that that may be part of what's happened in the very conservative write-up that we had before us. I would point out, as others have indicated, that cardiomyopathy, Schwann cell hyperplasia, and cardiac malignancies should be, in fact, combined for statistical significance, and that if you look, at realistic exposure scenarios, such as those we are experiencing in this room today, there are many of you that have in your pocket, on your body, a device that says, by the way, that it should be kept at least 5 millimeters away from the body, and you are all exposed to chemicals by virtue of being alive every day. And if we look finally at some trends in cancers, you will see some increases. The Ramazzini study that we talked about yesterday is now online. It is available. This is the report of the final results looking at the brain and heart tumors in the Sprague Dolly rats, which I would stress are the same animals here that have been studied. Again, this is a lifetime study, and the analysis of the levels you see here indicated. This is the Ramazzini levels. Over there on the far left, they are not visible in this current scale. This is what NTP exposed the animals to. This is the FCC public limits. This is ICNIRP. And here you have the occupational limits, which you can see are many times higher. And the French test at 10 grams SAR, you will hear from Dr. Arazi later today that when French government tested phones, over 9 out of 10 phones failed to meet the current standards and in fact exceeded the standards tested with a 10-gram volume. If you translate that into the US equivalent, this is how much radiation you are getting from your phones today. You are getting between 5 to 12 times more radiation than the government limits when phones are tested next to the body as opposed to the way they are tested by the government's test limits today. Now, this is just one table from Falcioni. I know we won't have time to go over it in detail, but I want to indicate to you, you see here the controls, and you see hyperplasia, endocardial, intramural, and schwannomas. But if you combine, this is now groups 2, 3, and 4, and if you combine the data on hyperplasia with the schwannomas, you find a statistically significant increase with schwannomas. It's not just the high-dose group, and we argue, of course, that you want to combine this. I actually visited the NovoCure laboratories, and I found not only, it's not a new technology, it's been developing since 2000. There are over 900 clinical centers that are now using it. It is considered by the Society of Neuro-Oncology as a state-of-the-art treatment for brain cancer today because of the fact that tumor treating fields can and will kill glioblastoma cells. What we see here in the results from the NTP is if you combine the data on heart, brain, and together, and you look at what Falcioni has done, you see that there is a combined effect, elevation of significant in male and female rats combined. And I think we have to make the analysis where you're not just looking at the schwannoma alone, but at the hyperplasias. And just to go back, if you look at the hyperplasia and the endocardial and intramural schwannomas combined, you get much more robust numbers. Now, the non-statistically significant dose-dependent increase is shown in the Falcioni data. And in a period over 20 years, historical control animals have had very few cases. But again, I stress that in both of these studies, it's important to use not the historical controls, but the controls in those studies. The conclusions of the Ramazzini study is that there are significant increases in the same types of tumors that near-field exposures of the NTP found with these far-field exposures from the Ramazzini studies. Of course, you will need more time to look at that, and we can't go into detail, but I think it's worthwhile to note that reproductive endpoints which were not seen in the study in terms of statistical significance of the NTP, actually do receive the highest dose. This is a simulation using some of the technology developed by ITIS with our colleague Claudio Fernandez, and you will see that the highest exposure when a phone is in the pocket to a male occurs in the gonadal zone, the testis. And this was a study done of a laptop without heat, showing the increase in damage into DNA and DNA fragmentation of the mitochondria of the sperm and sperm fragmentation. Of course, these slides will be available for your closer inspection. These are studies that my colleagues and I have done at Turkey, where I'm also on the faculty of Onda Kuzmayuz University, showing a reactive oxygen species in liver, kidney, hypothalamus, and testis. Again, statistically significant damage evident with these exposures. These are studies, again, on endocrine functions in uterine oxidative stress in 4 generations of rats, which you all know doesn't take that long to get 4 generations. And just 60 minutes a day, 5 times a week, results in a significant suppression of prolactin, progesterone, and estrogen, and increased oxidative stress. So what we have are non-monotonic reductions. This is now the NTP. I have taken data from table H2 in the current draft. And you will see here that in that draft, there's epididymal spermatozoal measurements. And I believe that, and I would like to talk to the statisticians about why they didn't apply a Williams test to these data, because I believe there is a statistically significant reduction in spermatid and spermatozoa in the NTP study. And again, you can't be all things to all people when we know that, but I would urge you to reanalyze those data in your study. These are data just developed yesterday showing pituitary cancers in the Central Brain Tumor Registry of the United States. These are rates, in the United States of one of the only national registries that we have of brain tumors of all types. And you see here a very real increase, and this is the age-adjusted rate of pituitary cancers as reported by CB Trust, which gets its data from the CDC. I think it would be a mistake to take anything other than this is a trend that should not be ignored, but it is consistent with what the NTP has also observed, which is an increase in these same cancers. Finally, I leave you this to look at YouTube, as you will see that the 5G, which is this new technology, which is in fact weaker in power for the way it will be used in phones, is being used for non-lethal weapons, and there's entire websites of the Department of Defense explaining this use. And I think I will stop at this point, but leave you with this. The breast cancers that were referred to are multifocal primary tumors. We have 4 cases age 21 so far. This means multifocal, different primaries, all occurring under where the antennas of the phone were stored. Now, this is not a statistically significant study. It's not an epidemiologic study. It's a case series report. That's all we have. These 4 21-year-olds have no family history, negative for BRCA. There is no known risk factor, and they all have multifocal primary tumors. Their physicians have contacted me because of our concern and interest in this. And in the meantime, what we are doing, I'm not making this up. You can get this at Toys R Us. It's a teething rattle case for an iPhone. There are over 10,000 apps for infants and toddlers so that your baby can play with the phone and the apps. Now, children can learn to swipe before they can learn to talk or sit up or eat, right? These devices don't have to be tested for safety. And it says on the box, protect your phone from the baby. That's what it says. I think that the results of the study here suggest we need to do a better job on things like this. And I wish this was something that I made up as a prop, but I assure you that it's not. Thank you very much.
Thank you, Dr. Davis. Any questions for Dr. Davis from the panel?
And if any of you pathologists would like to help in this analysis of the breast cancer cases that we have, I would be delighted, because we're looking for whether we can find a pathognomonic signature that might be useful here, because they're so unusual in the presentation.
Thank you.
Niels?
I assume that you're aware that the slide you showed about the exposure is mixing oranges and bananas? That you
Yes.
compare whole body versus spatial peak, and if you compare it fairly, the NTP study would be conservative with respect to the exposure.
Thank you for that point. I absolutely agree with you, yes. The NTP is conservative for exposure, and you'll see from the next presentation why it is the case. Because the French have a national agency which actually tests phones. And thanks to the work of my courageous colleague, Dr. Marc Arazi, a physician who has traveled here all the way from Paris, you will see that what the French have shown is that when you test phones, if I may take a moment, the way phones are tested.
We're way over time. So.
I understand, but I think it's important for the panel to understand this. The way phones are tested today, they're tested in a holster away from the body. Now, nobody here besides somebody age 70 uses their phone in a holster anymore. I plead guilty to sometimes doing that. But that's how phones are tested, with a spacer that can be as much as a centimeter or more off the body. And the same thing for the head. There's something called the pinna. We call it the ear. And the pinna can be between 5 to 15 millimeters away from the brain, and it is allowed to receive the same radiation as the foot or the hand. Figure that one out. That's why in that drawing I leave you of my beautiful little granddaughter, I point to her ear having the amount of radiation as a foot or a hand. So we would agree, Niels, you have to come up with a better system of testing, because the one you first invented almost 20 years ago or so is out of date, because it's measuring the way phones are not used. And what the French will show you is when you measure phones next to the body, you're absolutely right. Thank you for that clarification, Niels. The NTP is conservative in the amount of exposure that it gave. Thank you.
Okay, thank you. We will move on to Mark Arazi.
That's it. Hi, I'm Dr. Marc Arazi. As a French physician, I spent the past year studying the consequences of electromagnetic fields on health. Our new organization, Alert Phone Gate, has been created as a public educational institution. People have the right to know how to buy and use mobile phones as safely as possible. We wrote with Dr. Deborah Davis, our partner in US, to the FCC, asking them to soon revise their standards for a better protection of human health. First of all, we would like to thank the scientific NTP team for the quality and interest of this study. Your choice to use ISR level is particularly interesting because in the scientific literature, few studies have been done in this regard. We find your result concerning the effects on many organs, heart, liver, pancreas, thyroid, particularly important is in understanding the risk related to radio frequencies waves on the whole body, not only on the brain. Our comments today, and Deborah talked about that, will be restricted to the question of exposure that were used on the NTP study and how they will relate to recently released data from the French government National Frequency Testing Agency. The reviewing of this data are at the origin of the international sanitary and industrial scandal called PhoneGate. In July 2016, the French National Agency for Health and Safety, NIEHS, published an important report entitled Exposure to Radiofrequency and Child Health. The report brought that 9 out of 10 mobile phones tested in 2015 by the French National Frequency Agency, ANFR, in contact with the body, show a SAR level higher than 2 watts by kilo, and 1 out of 4 a SAR level more than 4 watts by kilo. After several legal action of our part, an NFR published on March on its data site the complete report of 442 cell phones tested until 2017. And you just have a part, and you will have the website to go and see for all the measurement we have. This result shows that the leading mobile phones sold in France, as well as Europe, as well as in 115 countries in the world, exceed the regulatory limits. All this was made possible by inappropriate regulation that allowed mobile phones to be tested at a distance of 15 to 25 millimeters from the skin. And just yesterday, a committee of 40 scientific experts of the French agency NIEHS confirmed in a new report that electrosensitivity is a real illness touching nearly 5% of the population, 3 million of French people. It is urgent and necessary to verify if there is a link between this overexposure of cell phone during more than 20 years and the appearance of this sickness. In June 2016, at the initiative of French government, the European Commission adopted a cautionary notice for manufacturers asking them to test the SAR extremities to 0 millimeters and the SAR trunk at a few millimeters without any upper limit. At this moment, it's 5 millimeters. And you know that US limit now measure for body SAR, it's 10 millimeters. And you can also say that Apple already, since 2 years, measure test at 5 millimeters. Moreover, as you know, the European standards are less strict than the American standard adopted by the FCC in 1996. If you take into account only the differences between this one. Okay. Between 1 gram and 10 gram to calculate the SAR, a mobile phone tested at 7 watts by kilo by NFR would have a SAR almost 3 times higher. It's going to be 20 watts by kilo for 1 gram of tissue and nearly 50 times the legal threshold allowed in the US. There is also the question of exposure time, both in the calculation of the SAR and in the choice for this study of alternating 10 minutes of exposure and 10 minutes without exposure. It seems really necessary, and I insist about that, to focus on realistic use made by users. So, if indeed it is difficult to compare the whole body SAR levels of your study and those of the SAR body and extremities, that is no more possible to say that humans are less exposed than rats and mice. So, I am here, after traveling 4,000 miles, to ask Dr. Buscher, face-to-face, to clarify his comment, almost when he headed to the media, that he would not change anything for himself, for his children, nor his grandchildren, regarding their habits of mobile phone use. I hope. Would you make the same comments with now? You imagine the serious consequences in terms of public health, especially in delaying simple measure of protection for billions of users. I will finish with this really simple advice I hear the first time from Deborah, and I say it all the time now, and I hope you will do the same. Distance with your cell phone is your best friend. Okay, I repeat, distance with your cell phone is your best friend. Thanks, and take safe.
Thank you. Any questions from the panel? Okay, thank you.
Thank you.
We will move on to Annie Sesko. You can please introduce yourself.
No. I did something wrong. I wanted to put the first slide on. Okay. Thank you. I'm glad to be here today. I'm listed as a private citizen. I will call myself a citizen of the world, yet I will add a few more things to this simple description because all... How do you get these things to advance? Done? Okay. Because I think I have been spending the last 35 years of my life trying to do something to prevent cancer in the world, and I think it has not been a big success. I am afraid to see that in the past 30 years, the number of cancer cases doubled in the world, and whatever I could do as French NIEHS researcher, or even during the 22 years I worked at the International Agency for Research on Cancer, which is part of World Health Organization, including 9 years as a unit chief of epidemiology for cancer prevention, what I really wanted to do, 2 years as acting chief for a program for cancer control of WHO. Things have not really improved. Of course, we have seen cancer increases because the population increased in the world, that we know, we can account for it, that it's aging, that's fine, we also know to account for it. But even when you look at childhood cancer, it keeps on increasing. And then I tend to be convinced more and more that when we look for causes of cancer, including in children, we really have to take seriously the environment we live in. And that has changed in a huge amount in the past 50 years, in terms of chemical carcinogen, but also in terms of physical agents. And I think EMF, ELF, all these fields have exploded. We are now living in a place, on a planet, where soon there will not be anyone unexposed, which will make it difficult, in fact, to demonstrate the fact that EMF exposure is a carcinogen. How is epidemiology going to manage when everyone will be exposed? Of course, we will try to do it in a more detailed way with the issue of dose response, which maybe will be providing some information. But I wish good luck to the epidemiologists of today for being able to conduct, for example, cohort studies of human subjects with unexposed. I don't know where they are going to find the unexposed. In terms of carcinoma, you have to rely with study. I know often present, for example, increase comparing a field in epidemiology. Most of the interesting results come from case control study. I know the results are very often presented at no overall increase, comparing exposed to unexposed, but I think we really have to focus on the only group of interest in terms of carcinogenicity, which are the ones who have been the most heavily exposed, either in terms of intensity of exposure, but most importantly, in terms of duration. And there, almost all case reference studies which have been done on this topic have found statistically significant, for most of them, increased risk. That's the case of the Nordic studies done by Ardell and his colleagues. That's the case of the interphone study for people who were exposed more than 1,600 something hours. But it's also the case of some studies presented as negative. For example, the cephalo study done on children aged 7 to 19 with brain tumors. And even there, the results are not statistically significant except for one, the most heavily exposed, but all the effect measures are greater than 1, so the risk exists. And we should not ignore that. So if we cannot do epidemiology, then we will be left with experimental study. And that's why I'm very happy to comment here the NTP study. There you can have an exposed group, and in fact one of my comments to the NTP study is that maybe the unexposed groups should be larger to avoid the use of historical control, but also from a statistical point of view, they should be larger than each of the exposed group, and maybe that will provide somewhat more stable results. So I just wanted to put a slide I got this morning from the UK. It's an in-press study looking at descriptive epidemiology, because if we cannot do etiological studies looking for the causes of cancer in humans, we will be able to look and follow up the statistics. And that comes from the UK. The dark graph, one going up, is glioblastoma. That's the worst form of brain cancer, the most aggressive, and it's going up from 75 to nowadays. Do we really want to see it keeping going up? It will. Nothing is being done. It will take a long while if we take preventive measure to make it going down. Of course, it's only a correlation time period, so that doesn't prove anything. But on the other hand, I think at least the question has to be asked. Same thing for pituitary tumors in Sweden. That comes from Ardell. And to get back and finish on the NTP study, I think it's really excellent that this study has been done, because it's a very large, well-conducted, methodologically sound study. No complaint about all of that. Of course, some questions may remain on the choice of exposure. And one point I want to make is that if we now have to rely on experimental study for the future, then we should really hurry up when there is a new technology being launched to do this kind of study. The question of a 5G, are we going to expose the whole population before testing it in animals in a careful way? Experimental study can provide some answers in 2 years. I know 2 years nowadays, it's a century. But nevertheless, if that could avoid unnecessary exposures to be put on the market, it will be good for the health of the populations. The differences, the fact that there are differences between rats and mice, males and females, some things which are statistically significant and other not, it has to be taken into consideration. But I prefer to look at the fact of the consistency of the result in between what we know from clinicians, from clinical studies, from EPI, from what we see in the mice and in the rat, in different studies. All studies have limitations, but when they keep on pointing in the same direction, we really have to take it into account. So I think that the NTP study provides more evidence of carcinogenicity. The Ramazzini also adds something more. And we need to have more on the newer frequencies which are going to be used. And it would have been so great to have it before the world population is going to be exposed to it. Why am I worried about that? Well, because I'm a doctor, after all, and I never forgot the Hippocratic Oath. First, do no harm. Primum non nocere. And I think just as a person, whoever you are, I mean, all people, including the industrial people who produce cell phones or whatever else, do not want to do harm to people. So we really have to take into account even small sentinels indicator. 10 years ago, with David Savon-Schreiber, who's now dead, we wrote an appeal. It was one of the first ones, and there were many more, saying to people, be cautious when using your cell phone. And in fact, I could have the same answer that, no, I didn't change my way of using a phone over the years, because already 10 years ago, we were telling people, keep the phone away from your body, try not to use it. If you have access to a landline, use the landline, text rather than talk. So there it was, and we already thought at that time we had enough data. 10 years later, I'm afraid to see so many more data has accumulated, and we still have to repeat these simple things. So now I tend to think precaution is even past. We should really enter into prevention. Thank you.
Thank you. Questions for Dr. Sesto? Thank you. Next we have Kevin Modis.
Could I just make one comment?
Oh, I'm sorry. I just.
It's a side, actually. The suggestion that animals will be the only unexposed controls. That's not quite the case. Just like human beings, animals are being exposed as well.
Well, but in the experimental lab, you can isolate them from the EMF. So you can still, with Faraday cages and whatever, I'm not an experimentalist, so you can do it in a lab. And I don't want that the only humans to be unexposed will have to be put in a specific place. I don't even know where, not even in monasteries in Tibet, wherever. And now with the satellites communications, the whole population will have access to internet 100% of the time, even if we are in the middle of Sahara. So for humans, it will be hard. For animals, it can be built in as an experiment.
But in reality, though, animals, especially those we use in the laboratories, are bred.
Yeah.
And they are bred by human beings. If humans are exposed to radio frequency radiation in where they are, the animals, whether rats, mice, dog, or pigs, whatever you choose to use, I don't think anybody going to choose pig as an experimental subject, they have already been exposed to radio frequency radiation. That's the point I'm making. Obviously, what you're saying that you can, based on your protocol, divide it into two groups, so-called unexposed and exposed. In reality, as far as radio frequency is concerned, they are all exposed.
So I think that terminology of background exposure is going to be more appropriate, because I agree that probably not unexposed. But we're going to move on now, so thank you very much. Kevin Modis.
Okay. So hi, my name's Kevin Modis. I'm with the California Brain Tumor Association. And if people had questions regarding my previous comment, I can be reached at wirelesseffects@gmail.com, wirelesseffects@gmail.com. Today I'm not speaking on behalf of, but I would like to highlight the comments of Leonard Hardell, who were submitted to this committee. He's an oncologist, MD, PhD, and one of the leading scientists worldwide when it comes to wireless radiation and cancer, and it's his work that the Italian Supreme Court based their ruling on, that ruled that cell phones can cause brain cancer. And his full comments can be found at environment@cancer.com, environment@cancer.com, and I encourage people to share opportunities for grants to help support his ongoing valuable work. Dr. Hardell is a senior professor of oncology. He has more than 400 scientific papers to his credit. He's led a group of scientists doing a multitude of studies focused on cell phone radiation and brain cancer for more than 20 years. He is a Collegium Ramazzini Fellow. He's retired from the Orboro Hospital, and he's now working with the Environment and Cancer Foundation. I wanted to highlight the conclusion of his comments, which take into account the findings of the NTP study, but also take into consideration his group's work, along with other studies up to date in terms of animal and human studies that looked at radiofrequency radiation and cancer, also looked at known mechanisms for DNA damage. And his conclusions were the following, and I'm going to quote him: clear evidence of glioma, clear evidence of vesicular schwannoma, acoustic neuroma, some evidence of thyroid cancer, some evidence of multi-site carcinogen. And I quote, based on the IARC preamble of the monographs, RF radiation should be classified as group 1. The agent is carcinogenic to humans. And now back to really my comments. This is from me and not Dr. Hardell. We now have the mechanism of how wireless causes DNA damage, leading to various health effects, and people can look at Yemenko's 2016 study that looked at and had the consensus of 93 studies. We're seeing an epidemic in the 3 areas of the brain that get the most cell phone radiation: the cerebellum, the frontal lobe, and the temporal lobe. And we're seeing the human evidence of people developing brain tumors. And I want to bring people to the attention to what's happening right now. Right now, Senator John McCain is dying from a brain tumor, and he's one of our leading, our finest leaders. He's also known to have 2 cell towers on his property. And we also lost, unfortunately, Vice President Biden's son, Beau Biden, who died from a brain tumor over his ear. And importantly, he was running for governor when he died, and he could have been our president right now. So this has strong implications. And I also want to bring to attention that LeBron James had a salivary gland tumor, and the salivary gland nerve runs in front of the ear. And we're talking about probably the most able-bodied, healthy citizen in the world developing a salivary gland tumor in a site where Israel has studies showing an increase in salivary gland tumors related to cell phone use. So I believe we are literally microwave poisoning our population and allowing it to continue unbridled, and that the FDA should take strong, quick action to rein in the FCC that we know is dominated by the industry. Understand this: the FCC is currently authorizing the release of 4 times as much wireless spectrum as we have ever had, with the rollout of 5G and the Internet of Things, promising millions of new cell tower transmitters and multi-millions of smaller transmitters being located in anything you can imagine. And as was mentioned before, floating cell transmitters low over the skies from balloons, covering our planet from satellite. We're talking about a thorough maximization of exposure, and that this program needs to take a strong stand and force the hand of the FDA and others to put a brake on what the FCC is doing, because based on this study and the other studies and Professor Hardell's insistence that we are dealing with a class 1 human carcinogen, we should be minimizing it, not recklessly maximizing it as we are now. And again, not testing any of these new frequencies for non-thermal effects, which this study clearly showed, and the Ramazzini study also showed. Regarding electrosensitivity, let's call it what it really is: microwave sickness. And I was asked by one of the panelists, in 1972, people can just Google Naval Medical Research Institute 1972, they articulated 122 symptoms of microwave sickness from Navy men that were being exposed by radar on their ships, and these were healthy men. And they cited 6,000 studies looking at thermal and non-thermal effects, mostly focused on non-thermal effects. So when the FCC and many people I talk to say there's no evidence and it's inconclusive or we're not sure, that's the industry talking. The reality is we had this information 1972. And we continue to ignore it. The fact that we continue to ignore it and have this conversation, and that this study is so critically important, shows the power of the industry to ignore the science, to ignore the sick people, and to push on for profit not for the protection and public health of the people. And everyone should be alarmed because there's no one who's going to be exempt from this, as they exempt these transmitters from any type of environmental review and any type of look at non-thermal effects. Everyone should be alarmed because this is a public health crisis, not in the making, in reality right now, which they are going to make incredibly worse to a point of being horrific. And the fact that the FDA is not taking stronger action and continues to spin this is criminal, because we are dealing with the people who are literally dying from this, literally being driven out of their homes by electrosensitivity, losing everything they ever had. And it must stop now. We must take action, and we must stop just doing our job and start doing what's right for the sake of our country, for the sake of our world. And I don't think this is any exaggeration given the findings of the NTP study.
Thank you, Mr. Modis. Questions from the panel?
No questions, really? All right.
Okay, thank you.
Thank you.
Check for a moment to see if our speaker from South Korea is on the line. Yes, let's go ahead.
Can I use this one?
Yes, that works, Dr. On. Okay, Dr. On, we have your slides. Please go ahead, and please introduce yourself.
Okay. And next, please. Next slide, please.
We have it.
Dr.
Yeah.
On, you can go ahead. We see your slides with your picture.
Yes, I would like to introduce myself briefly. I am a neurosurgeon and working at the Aju University Hospital, and I have been doing animal study in this field for recent 10 years as a member of EMF Research Committee in NIEHS, Korea. Next slide. Next, please. And recently, a draft report was released in public in 2016, and several important comments were already appeared in this draft. Since then, those comments have been reviewed and discussed. Next, please. And two NTP technical reports on cell phone RF radiation was introduced, and including the one article. Next one. Next slide, please. Yeah. Because I am a clinician, I would like to start with a slide demonstrating the brain tumor classification, like as shown below. And primary brain tumor, among them the glioma is that is the most common primary brain tumor. As you can see, the red color box, and those are high-grade glial tumors. And those are including the anaplastic astrocytoma and glioblastoma. And this glioblastoma is most common, highly malignant primary brain tumor. You can see the lower red box, 8% of schwannoma is demonstrated here. Next slide, please. Yeah. And glioblastoma, this is most common and most malignant glial tumor. And the mean age is 50 to 60 years old, and more common in men. And this is very biologically aggressive tumor. And its growth pattern is like this, and spreading to the parenchyma. like that. And the median survival is up to 12 to 15 months. And so despite of aggressive treatment with the surgery and radiation and chemotherapy, these tumors are most often relatively fatal. So the tumor is very serious for humans. Next slide, please. And secondly, the schwannoma, if it's about 8% in brain tumor, if we will include the schwannoma in peripheral tumors, the incidence could be much higher than 8%. And then about the cardiac schwannoma, this is very, very rare, extremely rare. And until now, in worldwide, only 16 cases have been reported. And this low picture shows that the one case report from Korea was published in 2014. Next slide. So I would like to comment some points. One, firstly, the NTP study scale was the largest ever. And this study design was very reasonable to perform with two groups, including the sham exposed and the RF exposed group. However, sham exposed group is different from the case control group. It means the real control group. Therefore, the data from the historical control group could not be an alternative, replacing that from the sham exposed group. Next, please. And both glioma and schwannoma, including cardiac schwannoma, are tumors of the nervous system. So even though cardiac schwannoma is extremely rare in humans, the NTP study reports have drawn special attention to tumors of the nervous system. If lifespan RF exposure may cause increased incidence of tumors of the nervous system, regardless of statistical significance, we have to pay attention to the carcinogenic potential of RFR in humans. Next, please. And this NTP study was very well organized, so we agree that. And however, the well-organized trial had not guaranteed the success of the study, especially in vivo experiments. And the survival rate of the sham exposed group would be the most important drawback for this study. Therefore, finally, the post-NTP study would be necessary to draw a meaningful conclusion. Next one, please. Thank you for your kind attention.
Thank you, Dr.
Yeah.
Does anyone from the committee have any questions for Dr. Ahn? Okay, thank you very much. We will move on to Paul Haro from McGill.
Thank you very much. I am still a physicist, and I still teach graduate courses in toxicology and health effects of electromagnetism at McGill University. Page 13 of the RAD data can be reworked to group the results according to tissue types, brain and nervous tissues, heart, and other organs. This cell tissue type classification highlights that brain and nervous tissues show carcinogenic action at various stages in various locations within the body, particularly if one accepts hyperplasia as one of the stages of cancer development. The mechanisms of metabolic disruption by electromagnetic fields proceeds through the Lorentz force, interference with hydrogen bridges, and electron tunneling, and is understood down to the quantum mechanical level. So I really challenge the notion that there are no mechanisms to support this. From a very basic mechanism of action on free electrons and protons, EMFs have downstream effects that reflect the complexity of cell metabolism. Biological systems are very intricate, and the views of engineering are generally too simple to take that into account. EMFs cannot be expected to have dose responses that are similar to those of alkylating agents, because instead of destroying chemical structures, they interact with enzymes and restrict and disorganize metabolism. Therefore, observations of tumor incidents that are considered in your report as equivocal, because they fail to display a classic dose response, the most cases of schwannomas in the heart among the rats in the lowest RF exposure group, should be taken more seriously, because the agent at hand operates in a different way than expected traditional chemical toxicology. Our work has found that past given threshold of action, EMFs can, for some variables, have an almost flat response over 2 orders of magnitude. Soon after the work of Wertheimer in 1979 on ELF, a number of scientists documented the mechanisms of action of pulsed microwaves at the level of mitochondria. Aaron Sanders at Duke University pointed already in 1985 to microwave inhibition of mitochondrial electrons transport chain function of ATP production. I found this group, the surviving members of this group, 6 months ago, and I asked them, what impact did your work have? They said it was completely ignored then, and it continues to be completely ignored. Yet, these people have 80% of the mechanism that they understand to be working these phenomena. We pointed to the same oxidative phosphorylation inhibition associated with ELF magnetic fields, describing the impairment of ATP synthase and its downstream effects on AMPK alpha. The changes produced by fields were similar to those of oligomycin administered and of oxygen deprivation. When oxidative phosphorylation in mitochondria is inhibited at the level of ATP synthase, delta Pm, the mitochondrial membrane potential, is increased. A 7% increase in this potential increases ROS production by 70%. That's MEWA 2003. Further, enzymatic reactions, the dehydrogenases that depend on deep proton tunneling that Inagaki 2013 are modified, leading to difficulties in ROS detoxification that have been extensively documented by numerous authors, Yakamenko and Dazdag. The susceptibility of the brain to oxygen deprivation is well known. In the context of this project, where multiple observations surround the cells producing myelin, it should be remembered that myelin vesicles contain functional FOF1 ATP synthase and respiratory chain complexes and are able to conduct aerobic metabolism to support axonal energy demands. In myelin vesicles, FOF1 ATP synthase activity decreased in the presence of specific inhibitors such as oligomycin A, Rivera 2015, the substance that in our hands, Lee 2013, suppressed oxidative phosphorylation under very, very small ELF magnetic fields. Although some have claimed that exposures used in the NTP are high in comparison with human exposures, this criticism ignores the fact that in the study, 2 years of rodent life are meant to represent the full human lifespan. If anything, the study underestimates the effects, and I would expect more cancers, as well as other health problems, diabetes in particular, to manifest later on should one use rodents with 70-year longevity. Whether it is amplitude modulation, FM modulation, phase modulation, or pulse modulation, biology always finds a way to extract the data from the carrier. If you have doubts about phase modulation, you can review the work of Theodore Litovitz, who's now passed away at the Catholic University in Washington on ornithine decarboxylase. And the unfortunate equation that I want to leave with you is that this means that if you increase bandwidth, the chances of interferences and consequences to biological systems also increased. I also really like Dr. Lin's comment about the fact that we don't have any controls. And those rats that we think are controls are in fact exposed to ELF, who have effects that are comparable to microwaves as we use them. And there's also the problem of genetic drift imposed by all of this exposure. And for those who know about the mitochondrial and to nucleus interactions, there's a selection barrier there that is weakened by exposure to these fields that can lead the whole of the biota on this planet to essentially genetic drift that could be irreversible. So those are very, very serious consequences that are not even discussed in the literature. Thank you.
Thank you, Dr. Mortis. Questions from the panel? Okay, if none, we will move on to our last speaker, Dr. Ron Melnick.
Hello. I'm here representing the American public, and when the NTP conducts a study, it's done for the purpose of identifying hazards, potential human health hazards, and to provide information that can be used in risk assessment, which would be used to determine whether there is a significant risk and whether that risk should be lowered by lowering exposures. So what I want to talk about is the utility of the NTP data on cell phone radio frequency radiation for assessing health risks. But also, I want to go back and provide a little bit of the rationale for this study, since this began many years ago. Go down. Okay, in 2000, I was asked to take on the cell phone radio frequency radiation project. Didn't know where to begin. Somehow it led to a phone call to Perry Wilson at NIST. We started discussing reverberation chambers. This led to interactions with ITIS, and you heard yesterday the consequence of all that interaction in terms of the creation of the reverberation chambers, the determination of exposures, and the dosimetry. So from the other end, on the biology, want to look at the objective. The objective was, as with most NTP toxicity studies, to test the null hypothesis. And to test the null hypothesis, you have to challenge the null hypothesis. Now, at that time, people, physicists and engineers in particular, were saying cell phone radio frequency radiation at non-thermal or minimally thermal intensities is incapable of inducing adverse effects. There's not enough energy to break bonds. That's why it's non-ionizing. It can't cause DNA damage. And we didn't know a mechanism by which it could cause an adverse effect. So the purpose was, first of all, we got to challenge the null hypothesis. And if we identify adverse effects, provide the dose response data that can be used to assess potential health risks for any of these effects. So going back to then, how do we design this particular study? When NTP conducts a study, we don't want to expose animals to what humans are exposed to or can potentially exposed to, because we're dealing with small group size and trying to identify low-level risks, which might be 1 in 1,000, which you can't identify with an experimental group of 100 or 50 animals, or 1 in 10,000. So typically, the studies are done at higher exposures, characterize a dose response, and use that information to quantify risk. So with the cell phone radiation, we run into a different type of problem, because we can't expose to much higher, because we would induce heating effects. So we decided to conduct a thermal pilot study to determine what could animals tolerate and still be able to thermoregulate their body temperature with less than a 1 degree centigrade rise. Now, why did we pick 1 degree? The FCC's limit is based on a body temperature rise of 1 degree, it was estimated to be at 4 watts per kilogram. And in fact, when we began the study, we thought that was going to be the highest level, 4 watts per kilogram. From the 4 watt per kilogram, the FCC divided by 10 to come up with the occupational exposure of 0.4 watts per kilogram, and another factor of 5 to come up with the human. These are just cut points, and also identified spatial SARs of 1.6 watt per kilogram averaged over any 1 gram of tissue, and 8 watts per kilogram for occupational exposures. So if we're going to characterize risk, target organ risk, the important value is that 1.6 watts per kilogram. Because if we expose animals to a much lower level, such as the whole body exposure of humans, the dosimetry in the brain would be lower than is permissible by the FCC. And people would say, oh, you got a negative study? That's what we expected. So because we were limited by the heating effect, to challenge that null hypothesis, daily exposures were extended up to 9 hours per day. And there had been other studies in which animals were restrained. We did not want the animals to be restrained. And that's how we came up with a reverberation chamber. At that time, we were considering studying frequencies 900 and 1900 for both modulation, CDMA, and GSM. But early on, Niels Kuster's group provided their initial dosimetry model where they compared the organ-specific SAR to the whole body SAR and produced this kind of graph, and this was discussed a little bit yesterday. But you can see with the mouse at 900 megahertz, most of the absorbed dose is in the tail. Similarly for the rat at 1.9 gigahertz. That was not the objective of the study, to find out what happens to the rat or mouse tail. We were most interested in was the brain, because that had been defined earlier from IARC studies and some of the human studies. And you can see at that time we were looking at a minimally different SAR in the brain compared to the whole body. And as was mentioned yesterday, the fat is low. There are other organs that vary. So when you expose the whole body, we're not going to get a straight line across all tissues, but the particular one that we wanted the most emphasis was on the brain and not on the tail. Okay, so the results that are in the technical reports show, I think, quite clearly that the null hypothesis has been disproven. There were perinatal effects. There were dose-related reductions in pup birth weights with both modulations, and in weight gains of pups and dams during lactation. We've discussed a lot about the heart. There are proliferative lesions, which included both malignant schwannoma and Schwann cell hyperplasia in male rats exposed to GSM and CDMA. There's also cardiomyopathy in the right ventricle in both male and female rats with both modulations. This occurred early because there was a 14-week interim sacrifice. The effects were dose-related. There was increased severity with increasing exposure. And there were increased incidences and trends of DNA damage in the brains of rats and mice. Also, there were many equivocal conclusions. But the proliferative responses of both neoplasms and preneoplastic lesions with both GSM and CDMA indicate that this study has identified certain target organ effects which have been called equivocal, but they equivocal to some agencies, such as some of the regulatory agencies, means ignore. And I'm concerned that when we see a target organ effect, they cannot be ignored. Two of the target organs that have that equivocal name next to them that I want to focus on are the brain and the prostate. So the data that is available for the brain and with both the GSM and CDMA exposures show that there were gliomas with both exposures. There were also glial cell hyperplasias, which are part of the continuum of the process, the cancer process leading to adenoma and carcinoma. Is also, if we total these, there's a total proliferative effect with both GSM and CDMA. To me, there's some evidence going on that this is definitely a target organ effect that should not be ignored. In addition, on these bees, I looked at some of the individual animal tumor data, pathology data, and some of those glial cell hyperplasias were considered to be high severity, marked severity. So we're dealing with a severity issue on lesions. When there's marked severity, these are close to being indicated as a tumor. They're called glial cell hyperplasias at this point, but when you look at the total picture there, the brain is obviously a target organ and should not be dismissed. Now, in 2016, NTP reported that the brain hyperplastic lesions and neoplasms of the heart and brain are considered likely the result of whole body exposures to GSM or CDMA modulated RFR. But in the technical report, it says now there was some evidence of carcinogenic activity of GSM and CDMA modulated cell phone radiofrequency radiation in male spray dolly rats based on schwannomas in the heart. The incidence of malignant glioma in the brain may have been related, meaning equivocal, ignore, to GSM and modulated cell phones. So I ask, what is the new evidence that led to a different kind of conclusion? Switching a little bit to the other organ, the prostate gland. As far as I recall, NTP has never identified a prostate carcinogen. Dave Mark, I think, indicated yesterday that maybe they did identify one. This is, although the historical control data are something that we got to take with a grain of salt, it does indicate how often this shows up in studies. It's a rare tumor. You can see because the one or the two identified in this study make up the historical control rate here, that we're seeing adenoma carcinoma, 7.8% with a range of 0 to 2%. This is a very unusual finding. And when we look at hyperplasia, in all dose groups, there is an increase in epithelial hyperplasia, part of the pre-neoplastic lesion, with increasing severity with dose. And when we look at this in total, we're finding significant effects at the 3 watt per kilogram and at the 6 watt per kilogram. I contend that this is not just suggestive, this is evidence, convincing evidence of a target organ effect that should not be ignored. So you ask the question, what about survival differences between exposed and sham? That's a good question. But taking a look at some of the data, the CDMA 6 watt per kilogram group had the highest tumor incidence of all the groups. This, at week 93, this is the CDM 6 watt per kilogram compared to the control. At week 93, these were identical. Prior to that, the CDMA 6 watt per kilogram had a lower survival. It was not statistically significant over the course of the 2-year study. So another aspect, looking at, again, some of the individual animal pathology data, there were no glial cell hyperplasias or schwannomas in the control or sham group. A glial cell hyperplasia was detected in an exposed rat as early as week 58 of the study. And a heart schwannoma was detected as early as week 70 in exposed rats. Therefore, I contend that survival was sufficient to detect either the tumors or precancerous lesions in the brain and heart of the sham group. Now, there's some explanation that there was reduction in chronic progressive nephropathy. And when you look at the CPN severity score of 3.7, it's extremely high. In the 1990s, when NTP was conducting studies, severity scores for chronic progressive nephropathy was a cause of death that led the NTP to switch their diets from NIH 07 to NTP 2000 to reduce the protein. This kind of score is something which is much higher than was observed during the 1990s and is a score that you find for a renal toxicant. And that's what is reported in the controls. Why it's so high, I don't have an explanation, and maybe it will be provided.
Dr. Melnick, you need to wrap up here. We're way behind schedule.
Okay, I just got a couple more slides. I just wanted to hit one issue related to the mouse, because that's listed also as equivocal. There's lung alveolar bronchial tumors. The incidents show a significant trend, and they're outside the range, which is much larger number of animals for the B6C3F1. Also, the malignant fibrohistiocytoma has a value of 5.6%. Very rare. One of those came from this particular study. So of previous 500 animals, only one animal showed this particular lesion. I think this is showing some compelling evidence of an effect. In the epidemiology, which is important, the IARC, I was a member of the IARC panel, and I think a couple of others here were on that panel, classified RF radiation as possibly carcinogenic to humans. This was based largely on the Interphone study as well as the Hardell studies. The working group concluded that these elevations could not be dismissed by bias alone, as that a causal interpretation was possible. Interphone study published in 2011 said the highest level odds ratios could be due to chance, but also could be a causal effect. More recently, there was an analysis of the interphone component in Canada where they did not see a significant effect. After that there was a significant effect, it wasn't ruled, recall bias was not a factor in the elevated odds ratio for glioma. So on my last slide, I think the NTP study is so critically important because it's identified target organs for both tumors and the preneoplastic lesions, which are the heart, the brain, which included DNA damage, and I think the prostate gland, the mouse also. There's now strong concordance between rats and humans in cell types affected, glial and Schwann cells, by RF radiation. This strengthens the animal to human association. What happens with this data, or what should happen with this data, is that the FDA would be responsible for conducting the quantitative risk assessment. How can they do this? It's looking at the response rate, which is, I would say, tumors and preneoplastic lesions, as a function of tissue dosimetry, which is the absorb power times hours per day for the duration of exposure in animals, percentage of lifetime, and extrapolate this to the RF dosimetry in exposed humans. From that, they should be able to quantify the risk for a certain amount of exposure in humans and then determine whether that risk is significant, where EPA, for example, is 1 in a million, is where they judge their risk. Because even a small increase in cancer risk could have a serious health impact due to the widespread use of cell phones, being 300 million in the US and 5 billion worldwide. Thank you.
Thank you. Any questions to Dr. Melnick from the committee? Yes, Dr. Felter.
Susan Felter. So a quick, over here, quick clarifying question. The tables that you showed combining the hyperplasia with tumors for the brain and the prostate, and it looked like they were just added. Did you go back and look at the individual animal data to see if they were in separate animals so that that total incidence was actually reflective of the incidence of animals? Because you could have hyperplasia and a tumor in the same animal.
Yes, I did. And in fact, I didn't read one of my footnotes. Let's take a look at footnote D. So with D, one animal was diagnosed with both adenoma and hyperplasia S. I did check all of those others.
Thank you. Yes, Dr. Adler.
Rick Adler. Thanks very much, Dr. Melnick. How quickly can we get copies of your slides? Is one question I have. And second is, when you say some evidence, are you using the same criteria that the NTP uses when you express it that way?
I'm using NTP criteria for some. If I was using my own, I'd say compelling.
Any other questions?
Can the chair answer my first question?
Oh, I'm sorry.
Yes,
Can we get
we'll get those
slides?
done during lunch.
Thanks.
Dr. Lin?
Yeah, thank you very much. It was interesting that at the beginning you said basically you had designed this particular study before you retired. So if I may, I'd like to ask a couple of questions that's related to that. First of all, we had some discussions on the appropriateness of controls, sham control versus historical control. Could you shed some light as to why the decision was made at that point to use only one common control, and also knowing that the historical control is very different from the exposures, the sham controls, how the decision was made to go in that direction?
Okay, well, because we were comparing exposure groups to sham, they were all in the same large room. We felt it was feasible to conduct that exposure for both either CDMA or GSM against that sham. There was a space issue related to the facility in Chicago in terms of putting additional chambers. In hindsight, maybe it would have been better to have included additional sham control groups, but at that time we thought by increasing the number of animals per group to 90 that we would have sufficient power to be able to detect small effects. But remember, even with a group size of 90, you need somewhere between 6 and 7% increase over controls to be able to see an effect as being significant. With respect to the historical control, I think this group has beat it to death, and I agree that the historical control is difficult to work with in this particular case because these animals are housed so differently. They are in EMF shielded chambers, which is so totally different than all other NTP studies. But I use the historical control to get a sense, for example, when looking at that skin lesion, to see how rare a particular event has been within the NTP historical control database. I don't use it for a quantitative comparison, but I use it to give a perception of, are we dealing with a very rare tumor, or is this a common tumor, and how much of an elevation might there have been over a common tumor. With the mouse lung, the control group was actually quite similar to the historical control group from NTP's previous studies.
I understand that. Isn't it true that the exposure chamber was actually determined ahead of time before this was ever went into effect? Therefore, you already knew that this was going to be a shielded chamber that's going to be very different from what the animals, historical animals, was the experience with. So the difference that you mentioned, apparently you already knew that, but you made the decision anyways. And related to that, somewhat that is, you also mentioned that the space limitations. As I recall, the call for proposal had specified you have to have ample space. In fact, if you did not, that would not qualify you as a potential contractor for that proposal. So that does not seem to drive at this point, anyways.
They had the space for 21 chambers.
So what?
They had the space for 21 chambers.
So
yes.
was that enough?
Was that what?
the space for 21 chambers enough to conduct the experiments as required? Or as you just said now, a little while ago, said they did not have enough space. So which one is the case?
The head space for 21, the design did not include 2 sham controls for male rats, for female rats, and for mice, because we felt that with the 90 animal per group, there was going to be a sufficient number of animals to make comparisons between either a CDMM or GSM exposure to a control.
So the fact of the matter was the decision to use one common control had nothing to do with the space limitations.
Oh, we couldn't add it afterwards, once the award had been made.
I think we're going to bring this to a close. We had a lot of discussion about that yesterday, and I don't see a point in rehashing it now. We can discuss this at the end. Thank you, Dr. Melnick. We will stick to our schedule, which means we have a shortened lunch period. We will promptly resume here at 12:50. Okay? Thank you.